Okay. Bismillah, alhamdulillah, wa salatu wa salamu ala rasulillah, wa ala alihi wa ashabi wa man wala. It's with a great pleasure that I would like to welcome all of you on this training session organized by ASID. This ASID African School is organized in the preparation of the 7th ASID Congress, which will take place on October 1st and 2nd, 2021. And it will be unfortunately online during this coronavirus pandemic. But I hope that we have surely occasion to meet each other face-to-face. ASID is trying to raise awareness on PID and its management in Africa since long time. In early 2008, one of our strategies is to train and train again. So this is the justification of this current session. This one will target B-cell immunity through its comprehension. We all know that apart from cellular immunity, humoral immunity plays a critical role in the development, presentation, and management of PID. Immunology in general and clinical immunology is gaining in importance with the new coronavirus pandemic. During the coming 3-hour times, we will be receiving information on the following topics with our guest speakers. I would like at this moment to say thank you for all of them by accepting to join us to give you lesson on B-cell comprehension on PID. B-cell development will be covered by Mirjam Van der Burg. I apologize for the pronunciation. These ladies come from Netherlands, and classification of B-cell defects will be covered by Nahla Erwa, our president of ASID. She comes from Sudan. Pathogens in patients with B-cell defects will be done by Angela Migowa. She comes from Kenya. Diagnostics for patients with B-cell defect will be given by Shuayb Elkhalifa from UK, and case presentation and discussion will be conducted by Hiba Shendi from United Arab Emirates. And finally, management of patients with B-cell defect will be covered by Reem Elfeky, who came from Mosque, Egypt, but I think that she is working at UK. All of you know the key role of B-cell and antibody deficiency in PID. That's it. They are a very frequent presentation of PID among the patient all over the world, especially in Europe. Maybe you have the same thing in Africa. I hope that you will enjoy this program and will help us in the future to raise awareness on PID in Africa. I would like to wish you a very good and pleasant session. Shukran jazilan. Thank you for being with us. Thank you and thank you.
Good morning, everyone. Welcome to this session, and I would like to thank the organizers for organizing this school. And I would especially acknowledge them, I think, for the program they compiled, because I was asked to give a basic lecture about B-cell development. And in my view, it is really essential to understand B-cell deficiencies or immune deficiencies in general when you understand the immunity, the basic principles of the immune system. Because I think this is a drawing of Escher, that's an artist, and I like this very much when thinking about immunodeficiencies and immunology. Because if you see one hand as the basic immune system, it really draws the other into existence. And I think we can learn from each other. We can learn from the careful looking at the patients with primary immunodeficiencies, and that will give you a better understanding or more detailed understanding of the immune system, which is as a consequence also again necessary to understand the immune deficiencies. So this goes hand in hand in my view. So the topic of today are the B-cell deficiencies, and therefore I will start with some basic immunology about B-cell development, and I think it's all concentrated on this beautiful molecule, the immune globulin molecule, because this is the molecule that's taking the lead in the whole B-cell system. And I think it's important to understand how it's generated, how it's selected, modified, specified, and how the effector functions are. So during this presentation, I will give a kind of introduction of the largest part of the immune deficiencies. So the inborn errors of immunity, how they are now most often called by the IOIS. So the term PID is slowly replaced by EAE, but I think both terms are still used, which is it's fine. Then you can see in this distribution that the predominantly antibody disorder, so the blue part of the circle, is clearly the largest. So most patients with an immune deficiency have an antibody deficiency. And also in the other areas, the B cells can be impacted. So genetic defects on the immune system often directly or indirectly impact on B cell immunity. Now, the cells that are doing the job are the B cells. Here on the left part, you see a B cell. So how it's in the periphery. It's a cell with a large nucleus, and there's also some cytoplasm. Here on the right part, you see a plasma cell. So in the end, the B cell develops into a plasma cell, and the plasma secretes antibody. And if you see in the structure of the cell, the plasma cell is really equipped for the large production of a large amount of antibodies, because you see here all the structures required for the protein production. When we go to the basic structure of an antibody molecule, it's visualized here. You have two identical heavy chains and two identical light chains. And the light chain can be a kappa or a lambda. And you can also see that there are two domains. The variable domain indicates in pink, and the constant domain indicates in green. And the two chains are interconnected with disulfide bonds, both the two heavy chains as well as the interaction between the constant region of the heavy chain and the light chain. Both the constant region and the variable region have globulary structures with long sheets and also some circles around. Here's also the variable domain. I will go back in that later in more detail. You have here the sheets and here the CDRs. And CDR stands for complementary determining region. And the complementary determining region is the region of the variable part of the immunoglobulin molecule that's involved in recognition of the antigen. The other parts are the framework regions, and the framework, as it's in the word, it's really the frame of the structure of the immunoglobulin molecule. In the next slide, I visualize the antigen recognition in more detail. Here again, the immunoglobulin molecule, and here how the variable part is able to recognize the antigen. Here from the top, these are the heavy chain and the light chain and the CDR3 regions, the 1, 2, and 3. And if you look at the binding from the top, you can see the following. This is the antigen, and the epitope is actually the part of the antigen that's recognized by the antibody. Here in the lower part, you have the immunoglobulin heavy chain with the variable region, and here on top, the light chain. And then you can see that the CDR regions are really making connection with the antigen with 6 position. And it's important to imagine that every antibody is unique. So the antibody produced by one cell. Now, I think I don't have to tell you that you have different immunoglobulin isotypes. You have the IgM, IgD, IgG, E, and A. And the only thing that I changed is the constant region. The variable region can be the same from the cell. And then the constant region can be replaced by a system called class switch recombination, which I will tell later. And the reason why you have different constant region is that the constant regions are responsible for the functional activity. So all the different isotypes have different effector functions. And this is nicely illustrated in this table. You have different functions from antibody molecules: neutralization, opsonization, sensitization for NK cells, for mast cells, and activation of complements. I will go into these mechanisms in a bit more detail in the next slides. But this slide is to visualize that certain isotypes are more capable of performing certain functional activities, like complement activation for IgM. For IgG1 is important for opsonization, and here the sensitization of mast cells. Here on the right part, you should also indicate the different immunoglobulins, how they are distributed throughout the body. So IgA, of course, in the gestational tract. So the IgG is mainly present in peripheral blood. So this is also giving the the distribution of the immunoglobulins over the body. So what I indicated, there's different functions for specific antibodies. For example, neutralization. Neutralization is important for the neutralization of bacterial toxins. So in this way, the antibody can bind the toxin, forming these structures, and these structures can be ingested by macrophages and can destroy and remove the bacterial toxins. Another function is the opsonization. So bacterial in the extracellular space can be opsonized by immunoglobulins. So if you see here, the bacteria are really covered by the antibodies, and these opsonized bacteria are also ingested by macrophages and then can be removed and destroyed. Then the third option is the complement activation, that are bacteria in plasma. So here, again, as in the opsonization, the immunoglobulin molecules bind to the antigen on the bacteria, but then in the presence of complements, the complement system is activated. And the end of the cascade of the complement system is lysis by the membrane attack complex. So there's ingestion and then also killing of the bacteria and removal from the body. So these are three different mechanisms of the effective function of the immunoglobulin molecules. There's also another way that's called antibody-dependent cellular cytotoxicity. That's especially when antibodies bind antigens on the surface of target cells. And it can be tumor cells, but it can also be viral infected cells that present some of the viral antigen on the cell surface. So then you get binding of the antibodies to these target cells. And then these antibodies are recognized by the Fc receptors of NK cells. And these Fc receptors bind to these immunoglobulin molecules bound to the target cells. And then you get a cross-linking, and then you can also have a lytic synapse formation. And then the NK cell really degranulates and then induces a lytic process. And as a consequence, in this case, a tumor cell, but it can also be a virus-infected cells, the cells die by apoptosis. And then finally, we have the IgE, the sensitization of the mast cells. So in this case, the IgE is bound with this constant domain to the receptors of the mast cells. Then, in case these antibodies recognize with the variable part the antigen, the allergen in this case, so it's an antigen, but in case an allergen, then there is a reaction induced, and the end result is the release of mediators like histamine. And these are released from the mast cells, inducing the allergic reaction. So this is, in summary, the function of the immunoglobulin molecules in the body. Then I talked about the function of the immunoglobulin molecules. Then I will now continue with how are they produced. And then now they are produced by B cells. So what I will do now, I will go back to the basic principles of B cell development. And the B cell development starts in bone marrow from stem cells, and you can distinct two phases of B cell development. That's the first part. It's an antigen-independent differentiation in the bone marrow, and this is aimed for the production of B cells with a specific B cell receptor. And then you have an antigen-dependent maturation in the periphery, and also in the lymphoid organs like the spleen and the lymph nodes. And in this antigen-dependent phase is aimed for antigen recognition, B cell response, the formation of memory cells, and also the formation of plasma cells with antibody production. And here in yellow on the bottom, you see different processes. This is called VDJ recombination, which is involved in the generation of the molecule. And here you see somatic hypermutation and class switch recombination. So these are things happening at the DNA level of the B cells. And what I will do, I will now talk you through the processes of B cell development in bone marrow and B cell development in lymphoid organs in a stepwise way. So in bone marrow, precursor B cell differentiation takes place. In the beginning, you have a lymphoid progenitor. And a lymphoid progenitor means that it can become, the cell can become a lymphocyte. So during this phase, the cell can become an NK cell, but still a T cell, and it can go to the B cell lineage. So in this phase, the REC is already expressed, then you have the first rearrangements. And from the lymphoid progenitor, the cell can still go to the T cell lineage, but if you see this differentiation route, and then especially the POX5 molecule, that's a transcription factor, if that's expressed, then the lymphoid progenitor is really committed to the B cell lineage. So as soon as POX5 is expressed, then all other lineages are suppressed. So then a B cell is really a B cell and cannot become a T cell or an NK cell anymore. So this is really a crucial molecule in the B cell development. If it's really committed to the B cell lineage, then it differentiates into pro-B cells, pre-B1, pre-B2, and finally immature B cells. Here underneath you see a lot of CD markers. Now you know that immunologists are fond of CD markers, so there are really a lot. And you can use them in flow cytometry to make a distinction between the different B cells, different cell subsets, in this case, the precursor B cell subset. Now, for example, now everyone knows CD19, that's the most familiar B cell marker. Then it's negative at the beginning, and then only from the pre-B1 cell stage, CD19 is expressed. Another marker that's often used for B cells is CD20 in peripheral blood, but as you can see, it's only expressed on the latest stages of the B cell development. So CD19 is so-called an earlier B cell marker. So what happens first is the rearrangement of the immunoglobulin heavy chain with DJ rearrangement, and important molecules of this process are RAG, the recombination activating genes. So what happens during VDJ recombination? During VDJ recombinations, the immunoglobulin heavy chain locus is rearranged. Now, the immunoglobulin heavy chain locus is composed of many variable gene segments, also diversity D segments, and joining J segments. And after this, you have the constant region. So in the first step of VDJ recombination, you get recombination of a D to a J gene segments. And all the intervening DNA is deleted via an excision circle, a B cell receptor excision circle, or having the signal joint. In the next step, you have the rearrangement of one of the V regions to the pre-existing DJ. And then you have a VDJ exon formed, and then you get translation, transcription to the precursor IgH messenger RNA splicing, and then you have the IgH messenger RNA, which is subsequently translated and then expressed on the cell surface. It's important to note that this region where the gene segments are joined, that's called the junctional region. And it's not like Lego that the blocks are put on each other without difference. At the border of the junction, so between the D and the J, nucleotides are inserted and deleted. So that's both occurring between the DJ and also between the V and the DJ. So that implies that this region, the VDJ region, can be really variable. Also, when you use the same V, D, and J gene segments, you have a lot of variation in this region because nucleotides are deleted and randomly inserted by the enzyme TDT, terminal deoxynucleotide transferase. And this is especially the region, the CDR3 region, which was important for the antigen binding. So there's a lot of variation built into the system, not only by the different combination, but especially also by the junctional region diversity. This is the IgH molecules, and that's here visualized together with the light chain molecule. But in fact, in this early stage of B cell development, it is linked to the surrogate light chain, because the light chain caprolactam is not yet rearranged. In this phase of B cell development, you only have an IgH combined with a surrogate light chain with the composed of V/B and lambda 5 or 14.1 in human. Here, it's indicated here. You have the heavy chain and the surrogate light chain. And it's just being tested whether it's able to signal, because if you have this pre-B cell receptor on the cell surface and you get a signal, and what's exactly the signal is not really clear, but if you get a signal, that's really important for a certain process going on in early B cell development. You get signal transduction via CD39A, B, SIG, BTK, important immunoglobulin of immunodeficiency gene, PSA gamma, and SLP65, that's also blink, and then you get proliferation. That's important that the cell starts proliferating to have more of the B cells with the same heavy chain, and then you can later on have a different light chain linked to that. Now this process can continue because you also need to differentiate, so then you have the termination of the proliferation, which is SLP65 and BTK dependent, and then you have the light chain opening and the rearrangement taking place at the light chain. So if back to the system, what I told you, have the pro-B cell, you have the rearrangement at the heavy chain locus, then you have a pre-B1 cell, then you get the pre-B2 cell, because there the light chains are rearranged. At this point, you have the IgH checkpoint, and that's the signaling via the pre-B cell receptor. So this is really an important checkpoint in precursor B cell differentiation. If for whatever reason there's not a good pre-B cell receptor or not a good downstream signaling via the pre-B cell receptor, the differentiation is blocked. And that can be the case in patients with an antibody deficiency. Then the light chains are rearranged, and then the aim is for the production of an immunoglobulin molecule with a heavy and a light chain, and then you get a selection against autoreactivity, and it can be clonal or that the cell becomes anergic, so cannot respond, or you get receptor editing, so then you have again a light chain rearranged and with the second light chain while removing the first one to see whether the autoreactivity is prevented or the cell dies. And then if it passes the second checkpoint, the cell can continue to the periphery. So here, this is the bone marrow where the precursor B cell differentiation takes place, and then the cell enters the periphery first as transitional B cell, and then it can go to a naive cell. And during this phase, you also have some homeostatic proliferation. So then you have more of the same B cell. And you see here that IgM and IgG is expressed. These are expressed by alternative splicing of the same molecule. Now, this naive B cell is able to recognize antigen. And this is the first step in the B cell response in the periphery. So we are now in the right part of the B cell development, and the B cell receptor is able to recognize full antigen, unprocessed antigen. And what's happening there? So here it indicates, and the antigen can induce a signal via the B cell receptor, but to get a good response, you need to get a second signal, and the second signal goes via the CD19 complex. The CD19 complex is indicated here with the components CD19, CD21, CD81, and CD225. So if the antigen binds, you really need two signals for the B cell to become fully activated. And when it's activated, you can get differentiation here in the mold tissue. and then also they develop into plasma cells. This side, so in the spleen, the marginal zone, then you also get activation, and then you have an antigen-independent differentiation into the natural effector cells. So the cells remain IgM, IgD positive, and then they can differentiate into plasma cells, and then they secrete IgM protein. So this is via the splenic marginal zone. And the last option is a T-cell dependent immune response. Then that happens in the germinal center of the lymphoids, of the lymph nodes. And for this, you really need the interaction between the B and the T cells. Here, the B cells recognizes antigen, and you need an antigen-specific T cell to get help to induce the T cell dependent immune response. Now, this is already a large square, but there's really happening a lot, which is crucial for B cell development, and also a lot of processes happening on the molecular level. So that's how we'll go in more detail here. So the B cell recognizes the antigen, and then it gives a signal to the nucleus. What happens? The B cell receptor, together with the antigen, is ingested, and then the antigen is processed, and then the MHC molecules are loaded with the peptides derived from the antigens. And these MHC molecules with the antigen are expressed on the cell surface. So in fact, the B cell becomes now an antigen presenting cell. It is an antigen presenting cell, but here it's exposed its function. And then the MHC molecule with the peptide is presented to the T cell. With the T cell receptor, which is antigen specific, it recognizes this antigen. As a consequence, the T cell becomes activated, and activated T cell starts expressing CD40 ligand molecule, which is important for the interaction with the B cell, because the B cell expresses CD40, and also the T cell expresses certain cytokines. And this way, the B cell becomes further activated. So this T cell signal is called the second signal, and that's important for the T cell dependent B cell immune response. So this is the cognate BT interaction. And what happens, the B cells enters the germinal center in the lymph nodes. During in the follicles, and maybe you've seen these histologic pictures, you have two zones in the germinal center. You have the dark zone, and this dark zone, the B cells start proliferating. And during this proliferation process, the immunoglobulins get somatic hypermutations. And somatic hypermutations are mutations that are randomly induced in the variable part of the VDJ exon, so in the VDJ exon, and thereby the amino acids of the B cell receptor can change. This is called this somatic hypermutation. So that can go through several rounds, and then the dark zone is left by the B cell, and then it enters the light zone. So then it stops proliferating, and then the B cell receptor, which has been modified because of the somatic hypermutations, is kind of checked by the follicular dendritic cell. So the follicular dendritic cells also present the antigen to the B cell, and then the B cells in this stage are prone for apoptosis, so they die unless they are getting the rescue signal for the follicular dendritic cells. And then only those cells with the highest affinity for the antigen are selected. And this is called affinity selection or affinity maturation. So it's in fact survival of the fittest. So the B cells with the highest affinity for the antigen are selected, and they receive a survival signal, and the others die via apoptosis, because not every change is an improvement. And then the cell becomes in this stage, and then it again has an interaction with the T cell. And then the class switching can be induced, the class switch recombination. And the class switch recombination changes the effector part, so the constant region. And then the cell can become a plasma cell expressing IgG or IgA, or a memory B cell. And here, whether it becomes a memory B cell or a plasma cell is also dependent on the strength of the signal of the T cell. So this is only one slide, but there's happening so much during this process, this phase of B cell differentiation. So I will go in some more explanation for the several steps, but this is the basic principle. It enters as an activated B cell and it leaves as a memory or a plasma cell. And then the memory cell can re-enter this again. So this is not a once in a lifetime event of a B cell. It can go via several rounds of differentiation from the germinal center. So as I told you, you have the affinitation by the somatic height mutation of Ig genes. And then if there's no good reaction and recognition of the B cell anymore, then the B cell dies via apoptosis. And here you have the B cell cross-linking, and then you have the proliferation maturation. So here again, you have the immunoglobulin molecules. The somatic height mutations are induced here in variable regions, and the aim of somatic height mutation is higher affinity of the antibodies. Then the other process was called class switch recombination, and that changes the constant region. So replacing it by the alpha or the gamma or the epsilon, and thereby you change the effector function. Now, an important molecule for class switch recombination and somatic height mutation is AID. And AID is the molecule that induces kind of mutations or variations in the locus of the IgH locus, and thereby inducing double-strand breaks. So what happens? You have here the VDJ exon that was created in the precursor B cell differentiation. This is the display, the visualization of the constant region, and you see here all the constant elements, and they are preceded by so-called switch regions, switch mu, switch alpha, and here switch gamma. And in these regions can be targeted by AID. And here in this panel, you see it in more detail. So AID makes kind of lesions that result in single strand breaks. And because you have a lot of single strand breaks next to each other that are induced by AID in this switch region, you get double strand breaks. And these double strand breaks are required to break the switch regions. So these switch regions are cleaved, so you have really double strand breaks. Then you have a circular excision product because the intervening sequence is indeed deleted by a class switching circle. And then the open ends are joined together here in the switch junction. So in this way, an IgM can switch to IgA. Here's indicated a one-step class switch recombination, but you can imagine that in a second step, this switch region can also make a switching to IgG4, for example, because this is still present. It can never go back to previous regions because they are deleted, but the class switching can also be ongoing, so consecutive class switching. So this indicates that AID is crucial for inducing class switch recombination. And AID is not only important for inducing class switch recombination, but it's also important to induce somatic height mutation. So here it's indicated what happens at the molecular level, and in fact that's also similar what happens here. AID makes from a C a uracil. So you get a kind of mismatch. And this mismatch, then the enzyme removes this uracil, and you get APE to remove this A basic site. and then it can be repaired in different ways. And in this way, it leaves an N, so it means that it was a CG and it can be any nucleotide in the end. So what happens during somatic hypermutation? The AID makes mutations here in this V region, which is important for the creation of diversity. So this brings me to the end of the B-cell differentiation. What I've told you that it starts in the bone marrow from a stem cell to an immature B-cell with a B-cell receptor. And in the antigen-dependent maturation in the peripheral lymphoid organs, you get T-cell independent maturation into plasma cells and memory cells. And you get a T-cell dependent maturation. And then in the germinal center, the B cells are further processed and selected by inducing of somatic hypermutations in the variable region, which is together with a selection aimed for a higher affinity of the antibody, followed by class switch recombination to change this effector function. So that brings me to the end of the B-cell differentiation. I hope it helped you with a better understanding, and I'm convinced that these molecules that you heard in the basic B-cell development all come back in B-cell deficiencies. And as I explained at the beginning of my presentation, it's important to understand this process to better understand what's going on in the patients. Thank you for your attention.
Good morning, everyone, and I'm really happy to be here presenting a talk on the classification of B-cell defects, and that was very nicely preceded by the very well-formed and explained talk by Dr. Mirjam, which was just the right talk before mine. What I aim to do today is to give you a review of B-cell function, which has just been elaborated on by Dr. Van der Burg. And I'm going to just touch on the very basic mechanisms and remind you of the molecules that could be involved in B-cell activation and B-cell function. And I'm also going to combine that with an overview of the phenotype of the B-cell defects and the pathogenesis of those, combine all this to try and make sense of what the available B-cell defects are all about. So B-cell functions include obviously antibody production. This is the most important effector function of B-cells. And ideally, we need the B cell to produce a polyclonal, or the B cells to produce polyclonal and functional antibodies, which are not autoreactive. So we want the perfect antibody that can, B cells that can actually identify all or most of the organisms and the abnormal antigens that we face, and to be able to help our immune system to get rid of the harmful ones. And we want it to behave itself and not act against ourselves through allergic or autoimmune diseases or mechanisms. We also want the B cell to be functioning properly in antigen presentation. And we also want it to interact with its other colleagues functioning within the immune system. So a lot is needed of a B cell to function normally. And to do that, there are steps which have been elaborated on, and I'm just going to review them here. So first come first, we need antigen recognition, because that's going to be the primary stimulus to a naive B cell. That will set off a whole myriad of activation processes from the receptor on the surface downstream to the nucleus. And for this stage, or for this function, in most cases, T cell help will be required as well in the form of the second signal. Once this is nice and well going, the specific B cell, the B cell will form a clone, or it will proliferate to make a clone that will be capable, hopefully, of producing antibodies which are specific to the antigen to which it has had an encounter with. And the ultimate outcomes would be antibody secretion, isotype switching. So you will have IgM production at the beginning, and that can switch to other types of immunoglobulin, depending on where your AID enzyme touches on the target switch zones that Mirjam was talking about. And then we will have affinity maturation of the receptor. And we also need a B cell memory to be formed, so that we can make use of that in future encounters. So in a nutshell, that's what we hope to get. As for what happens, why do we need these antibodies as effector molecules? Those again were very nicely shown by Dr. Mirjam. So B cells need to happen. The antibodies are needed for neutralization of microbes and toxins. They are needed for opsonization and phagocytosis of the microbes. The B cells also produce antibodies which can mediate antibody-dependent cellular cytotoxicity using the NK cells. It can lead to phagocytosis of the microbes which are opsonized with the help of the complement. So this is complement mediated through the appropriate complement receptors on the heavy chain. And B cells also can act as inflammatory cells, and they can lyse microbes, and they can activate complement, they can secrete cytokines, they can do many things. So these are the main functions of the antibody that we wish to get out of a properly functioning B cell. While all the antibody functions are very well characterized and known, there are other functions for the B cell as an effector cell, which need to be better studied and characterized, because they will they kind of help in understanding the functions of the B cell in specific diseases, mediating, areas of personalized medicine and a better understanding of these kind of diseases. So the B cell can act positively or can mediate T helper 17 differentiation, and through the right set of or the wrong set of cytokines, it can also affect autoantibody production. It can affect the macrophage activation. It can affect the regulation of the immune system in general, and get the balance right between a humoral and a cellular immune response. To do all this, the B cell needs a huge number of molecules. Some of them are on the surface and some of them are intracellular. Some of them are nuclear. And this is just how cells work. To the left here, we can see the BCR with the companion CD19 complex of molecules, while on the right-hand side, we see loads of surface molecules, including the MHC and all the co-stimulation molecules, where the immunological synapse forms with the T cell, and also all the downstream molecules that have been identified, and a lot more that are not shown here. There are also some of the enzymes that are needed for appropriate B cell development and appropriate B cell function. So let me just at this point mention that these are, there are loads of them, and any defect in any of these can lead to defective development, especially molecules that need to appear on the surface or are important for the development can lead to reduced numbers of B cells, maybe because they are reduced in number or because we can't identify them the normal way we can identify them. The other thing is defects in the intracellular and other nuclear molecules can lead to defective function. So a lot has happened with the advent of all the molecular techniques and with the genetic diagnostic techniques in the recent years, which have allowed an elaborate identification of such molecules and the explanation of the pathogenesis behind many diseases which were attributed to absence or malfunction of B cells. So B cell defects can have major presentations when there is an inherent B cell genetic defect. This may cause, as I said, a deficiency in the number or an abnormality in the function of the B cell. And such presentations can also happen when you have other molecules in the immune system. For example, if your T cell or your T helper cells are affected, you would automatically expect that your B cells will be affected in number or in function as well. As I also said, all of these defects result from defective development of the B cells. So they are not developing properly or they are not functioning properly. If we translate such defects into clinical presentations, we would expect to see patients who do not have antibody function, nor effector B function, or either of the two. So the clinical presentations would most commonly come in the form of infections, and these are most likely going to be recurrent respiratory, upper respiratory in the form of sinopulmonary infections, plus or minus other infections such as viral gastroenteritis, viral encephalitis, and many more. Functional defects can also lead to autoimmune manifestations, mainly in the form of autoimmune cytopenias and autoimmune endocrinopathies, as well as to allergic manifestations. And these are conditions that we commonly see in patients with conditions such as common variable immunodeficiency. And they collectively, autoimmune manifestations may take place in up to 30% of patients with primary immunodeficiency in general. Defective B cell function can also lead to problems with inflammation, and some of these patients may present with things like granulomas, such as the granulomatous disease or the sarcoid variant of CVID. And some patients may even have malignancies when they have abnormal or constitutive activation of certain molecules, such as the NF-kappa B constitutive defects, which may lead to such problems of malignancy. Lymphoma or gastric lymphoma is another problem that is encountered in certain groups of CVID patients. So the spectrum of clinical presentation is led by the infection in terms of frequency, or the majority of patients would present with infections which are either recurrent, persistent, unusual, or affecting mainly the respiratory tract, but also other things can happen. And there is often an overlap between the different kind of clinical presentations. And translation of such phenotypic presentations would be resulting from the antibody deficiency and the lack of the antibody function, the other abnormal B functions, which could affect the control of inflammation and autoreactivity and all that. And it can happen, obviously, either individually in the form of B cell defects, or it can happen when you have combined defects where the B cell is not functioning as a result of a problem with the other cells of the immune system, mainly the B cells. And these immunological manifestations or immunological findings that you may find, you could see in patients who have only the immunological disease of the B cell defect or the immunodeficiency or the inborn error of immunity, or such inborn errors of immunity can sometimes be associated with other genetic problems in syndromic patients, such as patients with ataxia telangiectasia and those with Wiskott-Aldrich, for example. Right. So B cell defects in general constitute more than 50% of all the inborn errors of immunity, and that's why we are focusing on inherent B cell genetic defects. And these are commonly named predominant antibody defects, and this is short or written as PAD or PAD. Let me just remind you once more of all the small molecules in the small boxes and the small circles before I move on to the next slide of the classification, because for the coming few slides, I'm going to talk about classifications of primary immunodeficiencies with special reference to B-cell defects. So why do we need to classify inborn errors of immunity or any other type of diseases, and how can we choose to classify them? So basically, any disease, you need to define it, and then you would like to have some sort of classification that will help you diagnose and to predict the course of the disease and the prognosis of the patient, and which will also guide the management of such patients. And how do we develop such classification systems? These usually, the best system would probably need to combine clinical presentations or clinical phenotypes and immunological phenotypes with pathogenesis and genetic defects. There are many examples that can do this. And I am presenting one of the simplest, and this is what we call the 4 main groups of primary immunodeficiencies, and these are what we use in our A project in Africa, in the basic medical level 1A projects. So this classification simply divides the primary immunodeficiencies into combined T and B cell immunodeficiencies, pure antibody deficiencies, innate deficiencies which include complement and phagocytes, and all the PID conditions associated with syndromes. And you will see, if you can focus on the top row, you can see that many of the classes that I will mention in the upcoming slides have been amalgamated in certain groups. Now, this is a classification which is very useful within a setting with very simple kind of diagnostic facility, because it relies mainly on clinical findings and basic immunological tests. And it also picks most of the PIDs, the most common PIDs with the major presentations. So in Africa, we tend to use this with 10 commandments or 10 recommendations, which are available on the ASID website. And using these alone, you can pick many PIDs, and you may even be able to name some of the diseases to a molecular level based on major clinical manifestations. However, it has the limitation of missing subtle defects and more complex presentations. It also, conflicts a lot with what's happening now with all the, the overlap and the genotype, phenotype correlations that do not happen in most of the diseases of the inborn errors of immunity. And in this slide, I've highlighted the table, the column, which we are going to focus on, to give you just a broad idea of what the clinical presentation of B-cell defects leading to antibody deficiencies would look like. So these would normally happen in patients who are more than 6 months of age, because by then the baby would have lost their maternal IgG and they would have developed an immune system, ideally, that is capable of producing specific antibodies. Obviously, this can be delayed a bit, so a patient might get proper immunoglobulins a bit later than 6 months. or they may get it a bit earlier than 6 months, but the average is 6 months. And these diseases may present up to the second or third decade of life. The aim of such a classification would also help in avoiding the undue delays in diagnosing such patients. It also defines the clinical presentation by suggesting that a patient who presents with 2 pneumonias within 1 year or who has bronchiectasis and or 4 new ear infections within 1 year and or recurrent diarrhea or skin infections may also qualify for this category of diseases. And then it also mentions the most common pathogens, which are the polysaccharide bacteria, the guardian. Obviously, the viruses are now coming up as major infective agents for antibody deficient patients. Diagnosis relies mainly on the finding of low immunoglobulins and on lymphocyte subsets to detect the number of B cells. So you have to, to try and get as much as information as possible regarding what the B cells of the patient are like and what is his or her antibody production is like. It outlines the treatment and the possible diseases or the most common diseases that fall under this category, including the A-gamma globulinemia, CVID, selective IgA deficiency, the symptomatic selective IgA deficiency, and the hyper-IgM syndromes other than the CD40 ligand. Now, as I said, this is a very simple way of classifying inborn errors of immunity or diseases of primary immunodeficiency. However, the internationally accepted way of classification has been developed by the IOS committee. The IOS stands for the International Union of Immunological Societies, and this is an expert committee which includes most of the international societies concerned with the subject. And the first meeting that happened for this, or that took place for this committee, took place in 1970 when they discussed 16 diseases, and they produced their first report in 1973. And since then, the report was updated every 2 years. The latest of these reports came up in 2019, following a 2019 meeting. And this now contains 416, and we believe that we are now talking about over 450 genetic defects following the latest update. As Mirjam has alluded in her talk, we are gradually replacing the term inborn errors of immunity, and that is gradually replacing primary immunodeficiencies. And the logic behind that is that it is not only the deficiency and it is not only the infection that results from such defects. Together with this classification comes a companion document, and this started appearing in the from the IOIS committee in the year 2013 rather. And this gives the phenotypic classification that describes the diseases that would be updated in the IOIS expert committee updates. And I would just like to draw your attention to the fact that we are now progressing a little bit from using the simple 4 columns of primary immunodeficiency groups to form or to develop African guidelines that are relevant to African environment and which comply with this IOIS classification. There is more detail to come on the main conference on the second day on that subject. So the current IUIS classification was published in November 2019. In November 2019, and it shows, it reflected that with all the improvement in genetic methods, there is an increase, a huge increase in the number of identified inborn errors of immunity. So from the year 1983, so that's 10 years after the beginning of the report. So in 1973, there were only 16 conditions. In 83, they became 50, and now in 2009, there are almost 450. There are 430 diseases, or 416 due to 416 defects, including 64 new diseases, as shown by the bars here on the right-hand side. If you concentrate here, you will see that table 1, which is the combined immunodeficiencies, and 2, the syndromic, and 3 is the B-cell defects, have got in between them the most number of increases, followed by the table number 6. And the way this document classifies B cells, or classifies inborn errors of immunity, as I said, kind of combines the genetic defects together with the pathogenesis and the clinical presentations. And this way, they created 10 tables of inborn errors of immunity groups, which share common pathogenesis. The tables go as follows. So the first one is for immunodeficiencies affecting cellular and humoral defects. The second one is for combined immunodeficiencies with associated syndromic features. The third one is for predominantly antibody deficiencies or B-cell defects. The fourth one includes diseases of immune dysregulation. The fifth one, congenital defects of phagocyte number and function. And the sixth one includes defects of intrinsic and innate immunity. The seventh one is the autoinflammatory disorders, and this is just growing very quickly. And the eighth one includes all the complement deficiencies. The ninth one is actually a new table which has been introduced in 2019, and this includes problems resulting from bone marrow failure and affecting immune cells. And also there is now a 10th table which describes phenocopies of inborn errors of immunity. So these are defects which result from phenocopies due to, for example, autoantibodies and to cytokines and things like that. And in fact, this table was table number 9, and it was introduced in the year 2015 as table 9. If we dissect these further, we will see that from what we have mentioned earlier and what Mirjam has mentioned as well, is that B-cell defects most of the time do not occur on their own because they need a lot of interaction with other cells, especially the T cells. So we will find some of the B-cell defects included in the tables number 1 and 2. So combined immunodeficiencies and combined immunodeficiencies with associated syndromes. But most of them and most of the inherent B-cell defects would be embedded in the table number 3, which is called the predominantly antibody deficiencies. If we also go to table number 5, about the congenital defects of phagocytosis, and those of number 6, intrinsic and innate defects, and 8, we will see that these 3 were actually embedded in the third column of the simple 4 diseases classification. So what I'm trying to say here is that this is an elaborate way of classifying the diseases, and I think the list will continue going on as long as we are getting better with genetic diagnosis. Because genes are teaching us a lot about our immune system, and the reverse is appropriate too. I'm also going to talk a little bit about the accompanying document that comes with the IOIS classification. And this is the document that gets produced based on the updated IOIS document. It is called the IOIS phenotypic classification. And the main objective of this document is that it tries to provide a user-friendly resource for clinicians at the bedside. So you see the patient and you think about which category based on a number of trees which are color-coded. And there is also these tables also, or these color trees include the disease names which are presented in red within the tables, and the genes behind them are in bold italic. So it combines photographic memory with pathogenesis as well as with the rest of the classification criteria. And it's also available in smartphone app, which can come in very useful at the bedside. And this is what I'm actually going to focus on for the coming 2 slides before I finish. So the table that includes, or the group that includes the B cell defects, which are inherent to B cells and which lead to predominant antibody deficiencies with hypogammaglobulinemia, are all included in table 3 in both documents. So, and the first one of these, so in this case, you will see a patient who is either having problems with the infections, the plus or minus the immunity, autoimmunity plus or minus the allergy. And then you investigate them, you find that they have low immunoglobulins. If that is the case, then you have to exclude secondary causes. In places like Africa, we need to exclude HIV and other kind of endemic diseases and things like that. And also we have to look very carefully and listen to the patient very carefully, ask them questions to see if we can pinpoint a drug or a malignancy or, a problem in their urine or in their urinary tract or the GIT or skin that is causing them to lose their immunoglobulins. The next step would be to measure their B lymphocyte count. And then based on the information we get, we divide the patients into 2 groups, patients who have absent B cells. And these are mostly patients with problems that have affected the development of the B cell through genetic defects of enzymes, such as, for example, the BTK mutation. And the other group would be patients with low immunoglobulins and a B cell count of more than 1%. And in that case, you will on the plot or on the flow cytometer, you will see a proper population of B cells, but the number is less than 1% of the total lymphocyte count. And in this case, you will define a common variable immunodeficiency phenotype. Now, common variable immunodeficiency is a clinical entity, and we've been told since our early training days that this is like a an apple that is gradually being eaten, and each time one of the monogenetic diseases that comprised under or included in this group of diseases is taken away from the apple. So many genetic defects have been defined under this category, and many very interesting ones too, especially the advent of genetics and the definition of all the gain of function versus loss of function mutations. Many diseases have been identified, such as the APDS, for example. And if you remember that picture of all the molecules that a B cell may need, you will think of all the surface molecules that could be defective. Most of them are actually found here on this side, included under the CD19 complex defects. And many are functional intracellular molecules that are needed for downstream signaling. And such defects would affect the function. You may find that the patient has B cells, but these B cells are not able to do the functions that we've listed earlier. And the examples are many. And as I said, all the advance of genetics has just made things very interesting and exciting. And this is an area of great overlap as well with diseases of immune dysregulation and combined immunodeficiencies. And, you know, the line just sometimes becomes very thin where to place a patient. But these are the ones that have been identified as mainly primary antibody deficiencies, but the clinical presentation may encounter other or may have features of other kind of combined immunodeficiencies or so. So this is for patients with low immunoglobulins. So low immunoglobulins and absent B cells is one category. Low immunoglobulins with present B cells is all the common variable immunodeficiencies. Now, there is another table, which is table 3B of the predominantly antibody deficiencies, which includes all the other antibody deficiencies. These are all lumped together in this table without specific classification of what the B cell numbers are like, but in most cases, the B cells are present in this category. And there are three main groups. So you have a group with severe reduction in serum IgG and IgA with normal or elevated IgM and normal numbers of B cells. And this includes all of the, or most of the hyper IgM syndromes. And you will see here that they include AID deficiency. I hope you still remember that enzyme, and the ONC deficiency, which also has been demonstrated beautifully by Mirjam, and the Iono80 and the MSH6, but they do not include the CD40 ligand, which can have a similar presentation, but falls nicely under the combined immunodeficiency group of diseases in table number 1. Now, isotype, the other group here includes all diseases with isotype or light chain or functional deficiencies. So patients who cannot isotype switch or have properly functioning light chain combinations or other functional deficiencies with normalish or normal B cell functions. And these include all of these gray diseases that have led to a lot of controversies, including the selective IgA deficiencies, which could be symptomatic or asymptomatic, the transient hypogamma immunoglobulinemia of infancy, which sometimes is considered a disease of maturation of the immune system, but more and more is coming on that. IgG class deficiency combined with IgA deficiency, isolated IgA subclass deficiency and specific antibody deficiency with normal IgA and normal B cells. So many of the unknown diseases, but the ones that we talk about a lot are included within this table. And I think some of these will gradually go into the monogenetic common variable immunodeficiency very soon. Some of them might go to the combined defects. So there are many of these in this group. And the third main group of this one is the high B cell numbers. Is a group of patients who present with high B cell numbers due to constitutive NF-kappa B activation. And this happens in problems such as defects or deficient or genetic defects of the CARD19 gain of function mutation, where there is a lot of activation of B cell proliferation, not necessarily properly functioning. But these are the group of patients who may have issues with poor vaccine responses because the function is not right, but they can also have problems with malignancies. Now, with this, I hope I could review the major functions of the B cells and their defects. And I hope that I managed to make sense of how these could be combined with pathogenetic mechanisms and phenotypic characteristics to define classes within the IUIS classification, as well as with the simpler type of classifications to define B cell defects to help us clinicians in diagnosing and predicting prognosis for these patients and helping their management. I also hope I did a reasonable overview of the IUIS and phenotypic classification and explained the presence of the two documents, because I think these are really crucial documents for anyone who would like to, have a reference on B cell defects. So with this, I would like to conclude and thank you very much for your attention. Thank you.
Good morning, everybody. And I would like to really thank the organizers of this session for giving me the chance to present in this esteemed Congress workshop. And in a special way, I want to honor each and every one of you who have logged on. So I'm Dr. Angela Migowa, a pediatric rheumatologist from Kenya. And after those phenomenal talks by Dr. Mirjam and Dr. Nahla, I will now shift gear into more of a clinical approach to look at the pathogens in patients with B-cell defects. I'll begin with a quote by the Nobel Peace Prize winner from my country, who said that you cannot enslave a mind that knows itself, that values itself, that understands itself. And I really want to thank the African Society of Immunodeficiencies for this great advocacy to help us understand better primary immunodeficiencies so we can improve the lives of our patients with these entities. So the objective of my session is to highlight the various pathogen infections that we are likely to see in patients with B-cell defects. And this shall be the outline. We'll have one slide on the overview of the function. Dr. Mirjam did a great job at that. And then another one slide just on overview of the immunodeficiencies, just the B-cell dysfunction. Again, Dr. Nahla did a phenomenal job at that. And then I will spend much more time on the history, clinical features to look out for, to suspect patients with B-cell dysfunction, and what are the pathogens that we're likely to come across. And I think at the end of the entire workshop, we then have questions and summarize. So I remember in my days as I was doing my undergraduate medical training, there was the perception that the humoral immunity was the main pillar of defense against bacterial infections. It still is so, but thought not to be very, very key in viral infections. And we shall see as we discuss that there's a paradigm shift in our understanding of the role of humoral immunity. B cells are key players in the adaptive immune system, fundamentally because they produce antibodies. And above all, they have the capacity to maintain the memory of pathogens they have been exposed to, so that in the future, if there's another re-exposure, then they develop a rapid and stronger response. However, we always have to keep in mind that nature has a way of maintaining a delicate balance between the B cells responsible for recognition of pathogens and appropriate response, vis-a-vis having B cells that attack the self, B cells that attack the individual. And that's where we as rheumatologists come in, because that's our bread and butter, dealing with the autoreactive B cells. But if all else is well, there's normally a delicate balance between these two concepts. And Dr. Mirjam highlighted this as she was going through the various actions or roles of the B cells. And I think here it's important to emphasize that by the virtue of the fact that the B cells produce antibodies, their neutralizing function in mopping up pathogens before they gain entry into the cell is very key. However, if the pathogen then gains entry into the cell, then of course various other immune defense systems eventually come into play. But this concept of mopping up pathogens is very key also when you're trying to combat viral infections. So I've borrowed a tagline from people who work in the defense forces, where they say the best defense is a good offense, and our B cells are fundamental in offering a good offense. So this is just a very simple schematic overview of what Dr. Mirjam spent her session on, just to explain that, when you have the naive B cells, upon exposure to a pathogen, they would move to germinal centers, proliferate, and you'd eventually have either long-lived memory B cells, long-lived plasma cells, or short-lived plasma cells. But all this doesn't exist in isolation. There's always a check and balance, and that's when you've got your regulatory B cells that come into play. And of course, all this is mediated by cytokines and other chemokines responsible for this process. And Dr. Nahla already has gone through, what are the various potential inborn errors of immunity that you might see that are unique to B cells, so and so to speak. And she's done a great job at this. For example, if you have got defects in the Bruton tyrosine kinase, then you'd have X-linked agammaglobulinemia, for example. If you have got defects in B cell survival, then you'd have, for example, examples of common variable immunodeficiencies. You could have defects in B cell migration and activation. And an example in this is Wiskott-Aldrich syndrome. Or, again, Mirjam went into detail as to, the class switching process. And you could see it was not, it's a complex, it's an intricate process that takes place. So if things are not right in that intricate, complex process, then of course you can have other entities like common variable immunodeficiencies, things like hyper-IgM syndrome, ataxia telangiectasia, and then you could also have defects in the maturation of some of your B cells and all the other molecules that they would produce, and then that also can present as common variable immunodeficiencies. The highlight of this is not to try and remember each and nitty-gritty detail, but to be cognizant that you could have defects when you're along the maturation phase of the B cell, defects that are important for B cell survival, defects in the migration activation, defects in class switching, and maturation defects. So what are some of the pertinent things to keep in mind? If you were to encounter a patient, what would you hear, what would you see that would make you suspect that this is someone who potentially has a B cell defect? And this is really key, because it has been found that sometimes the reasons why we don't pick these cases is because we don't even suspect them in the first place. So I have got a number of tables to share, and I apologize if it looks like it's a busy slide. But nothing will ever replace a thorough history and clinical examination to help you suspect these cases. And with your suspicion, then you will then institute the necessary investigations to confirm your diagnosis. So in the history, the infection history is very key. And you're going to be looking for infections that are not common. For example, a child who might not have, HIV, but is presented with pneumocystis pneumonia, you're going to wonder what else is going on. You're going to wonder about the severity of the infection. For example, a child who might have a pneumonia or otitis media, which responds to coamoxiclav originally, but all of a sudden, you have to scale up antibiotics to include ceftriaxone, meropenem, peracillin, tazobactam. You need to ask yourself, why is such a severe infection needing so much antibiotics of very high titer antibiotics? Or if there's a history of abscesses in the internal organs, subcutaneous abscesses, prolonged, protracted diarrhea, as I said, infections with unexpected opportunistic infections, extensive candidiasis and oral thrush, molluscum contagiosum that keeps recurring. If you start hearing that kind of a infectious history, you need to ask yourself, is this a child or a patient with a potential inborn error of immunity? And we shall see later how you could suspect when it is potentially because of a B cell defect. Family history is paramount. And here you're looking for any unexplained infant deaths that are due to infection, especially adverse outcomes after vaccinations. That is very key. Sometimes it's ignored, but it's an important red flag. You're looking for consanguinity. And for us rheumatologists, when we start seeing autoimmune diseases that are outside the typical epidemiological age range, then we wonder about inborn errors of immunity. So for example, we'll take the example of lupus. It's not very common to find a 2-year-old child presenting with the classical clinical syndrome of lupus. So for us, if we ever encounter that, we begin thinking of other inborn errors of immunity as the primary reason for that presentation. And then, of course, there are other issues that you could detect in the history that could point you towards suspecting patients with inborn errors of immunity. For example, delayed separation of the cord, delayed loss of primary teeth, for example, failure to thrive. That's a big one that's often overlooked, because nutrition also has an impact in affecting how your immune system functions. So anyway, looking out for failure to thrive, bleeding tendencies, angioedema, hemolysis, granulomas, peculiar inflammatory bowel disease, among others. When it comes to physical examination, what are you going to be looking for that could be potential red flags that we might be dealing with an inborn error of immunity, or the skin has a wealth of information? Look at the texture of the hair, distribution of the hair, color of the hair. Look at the dentition. All right. We know, for example, in hyper IgE syndrome, you've got double rows of dentition. So look at the dentition. Look at the nails for dystrophy. Look for any other peculiar pigmentation. And most of the time, I'm always stuck. I keep touching base with the dermatologists to guide me on what they think this peculiar pigmentation is about. So these are not patients that you're going to manage on your own in isolation. A lot of teamwork is important. We've talked about the oral cavity. You're going to look for oral ulcers, like I said, dental crowding, persistence of the deciduous teeth, or what sometimes is known as the primary teeth, enamel hypoplasia. You're going to look out for telangiectasia in the eyes, which you could see in this Scott Aldrich, for example. Looking even just for the lymph nodes, we're always looking into the throats of our children. Are there lymph nodes present? And I think I've had just one child whom I encountered who did not have any lymph nodes, a 2-3-year-old, which is very peculiar. Look for evidence of asplenia, for example. Neurologically, if you find a child with microcephaly, macrocephaly, and if there are other features in the examination and other features in the history, then you might wonder about that. I won't go so much into the investigations because I know that's going to come through, but sometimes you might stumble across a child who's been sent to you and somebody already requested investigations. And for me personally, the full hemogram or the full blood count is one of my favorite investigations because there's so much information there by looking at the differentials of the white cells, looking at the parameters of the RBCs, getting that report on about the peripheral blood film. And there are times we've picked children just by the red flag that was detected in the full hemogram. And of course, then other features that could be involved is hypocalcemia, hypotriglyceridemia, hyperferritinemia, and low CRP. There have been cases that I have encountered in my practice with my colleagues where a child comes with such terrible infection, very high fevers, and a low CRP. When we see that, then you wonder about an inborn error of immunity. I want to emphasize really the importance of pattern recognition. What are some of the things you might hear in the history? And this is not unique just to the B cells. We shall go into the B cell defects shortly. But, certain things you'll hear in the history or you will see on examination, and what are the likely pathogens you can postulate are implicated. Of course, we know the gold standard for some of these things is culture, but this might not always be available in all the 52 countries of our continent. So what are some of the things, if you hear and see, you should suspect, and what are the potential differentials? So a child or a patient who has got recurrent ENT and airway infections, all right? It is important, of course, you keep in mind that it's not something that happens like every month and doesn't need high-scale antibiotics, all right? But, some of the pathogens you should suspect are Haemophilus influenza, strep pneumonia, Moraxella, sometimes even staph. You need to consider other things that might come on board, like persistent bronchitis, bronchiectasis, and they could even complicate based on the pathogen with meningoencephalitis. The other things that you need to consider as a differential, because as clinicians, we are like investigators, so things like cystic fibrosis need to be ruled out. If they have been on any medications that are potentially immunosuppressive, you need to rule that out. And I'll say, by virtue of being a pediatrician, the issue of malnutrition also is very key. We've had some cases whereby they're very malnourished, but when you sort out the nutrition, all these recurrent infections actually resolve. So those are things that you need to think about, things like ciliary dyskinesia, for example, or alpha-1 antitrypsin deficiency. Secondly, you could have a child who just presents with failure to thrive right from infancy, you know, and they say that they keep feeding this child well. Some of the things that you need to keep in mind is that could this child be having viral infections that have not been sorted out, like CMV, VZV, HSV, even things like cryptococcus. And I'm assuming that these are patients who you've already done due diligence and ruled out infections such as HIV. Some of the features that could stand out in these children is that they have this intractable diarrhea, or they have infections with very peculiar organisms that are not typical for them. And you know, other things that you need to be worried about, and Dr. Nahla mentioned that, is that sometimes these are people who could evolve into malignancies because, you know, the immune system is responsible for keeping in control any abnormal multiplication of cells. Don't forget about the possibility of toxicity like lead poisoning that could lead to this kind of a clinical presentation. Then you could also have a situation where you have got recurrent pyogenic infections. You know, they are always coming, these pyogenic infections, and you need to wonder about phagocyte dysfunction. And some of the pathogens implicated in this kind of patients include Staphylococcus, E. coli, Enterobacter, Pseudomonas. Even you could have disseminated fungal infections sometimes in these children. And one of the things to rule out as a differential, of course, is, and we are culprits for that as rheumatologists, because we're always immunosuppressing our patients. So you need to make sure that there are no other secondary causes of that before you label them due to inborn errors of immunity. We've talked about the issue of unusual infections with pathogens like Cryptosporidium, Microsporidium, Histoplasma. And, you know, sometimes the other thing to look out for is that these atypical organisms don't even respond to the treatments that typically would clear this kind of infections. And then if you keep seeing recurrent infection with the same pathogen, you know, today it is Salmonella typhi, tomorrow again it's Salmonella typhi, again we are back at it at Salmonella typhi, you need to question that. And you know, some of the differentials that you need to consider, of course, will be occult infections with reservoirs, you know, hiding somewhere in the endocardium, for example, or they just had inadequate treatment. You know, the dosage of the antibiotic or the antimicrobial wasn't optimum. Then, of course, if you see children who have got autoimmune conditions, especially not in keeping within the typical epidemiological age bracket, we always think of inborn errors of immunity, and we consider that in the workup. Other times they could also present with angioedema, and in this case, like I said, this is not isolated to B cell defects, but ultimately you need to rule out other potential causes of inborn errors of immunity. So, you know, what are some of the starting points when you suspect? I say that blood count is very important. If you find that you're having low counts, and that depends, a lot depends on what you have within your environment, within your context that you can use and get quickly, because in these patients, more often than not, a prompt and early diagnosis with the right treatment and management is very key for their survival. So, you know, start with your blood counts, look at the differentials of your lymphocytes, then you can step up and look at the immunoglobulin profile of your patients. If you're in a position to actually assess response to vaccinations, then please go ahead and do so. And, you know, for example, if you're suspecting agammaglobulinemia, then you can proceed on and even do your B-cell enumeration and even CH50 tests as and deemed appropriate, but this shall be covered later. As I said, when I was opening my presentation, the perception of potential pathogens in patients who have got B-cell defects was typically thought to be mainly encapsulated pathogens and gram-negative bacteria. But now we know, as was highlighted by the two speakers a priori, that the B cells are also very important for protection against viral infections. And here I've just picked like 4 examples of patients who potentially have B-cell defects. Good syndrome, I think we've talked about CVID and X-linked agammaglobulinemia. Good syndrome is a combination of a thymoma, myasthenia gravis, and immunodeficiencies. We currently have a child we are working up for this in my unit. And we as rheumatologists are very notorious for, rituximab. We give rituximab, and this, of course, post-rituximab, you can have patients who come down with quite severe viral infections. So you can see across these 4 different phenotypic presentations of B-cell defects, you could have patients who, after enteroviral infection, across the board, they could present with meningoencephalitis. You can have these patients succumb to Coxsackie virus infection, and they can present with arthritis and gastroenteritis. You could also have the JC virus, adenovirus, and you can see, like for adenovirus, they could present with a dermatomyositis-like syndrome, which is in our domain as a rheumatologist, or even a chronic lower respiratory tract infection, or even fulminant hepatitis, as in the case of anti-CD20 therapy. In the cases of infection with mumps, measles, and rubella, you could see fundamentally they could present to you with an encephalitis-like picture or a fetal pneumonitis. When it comes to herpes simplex virus, they could also present with an encephalitis-like picture, an ulcerative dermatitis, tracheitis. The same goes for varicella zoster infection and CMV. CMV also very notorious for giving you chronic diarrhea, can give you a retinitis, can actually give you everything, infects you head to toe. I want to talk a little bit more about SARS-CoV-2 because, we're amidst this pandemic, and you can see that patients with B-cell defects of varying degrees are at risk of persistent and severe pneumonia. And we're going to look at some data to show how patients with B-cell defects in the face of COVID-19 infection have poorer outcomes. So what are some of the peculiarities in some of these various phenotypic presentations of the B-cell defect? In X-linked agammaglobulinemia, for example, it has been found that children who would have this condition have developed vaccine-derived poliovirus infection in comparison to other primary immunodeficiency disorders. Why I'm highlighting this is to dispel the myth that B-cell defects mainly affect mainly affect bacteria, particularly encapsulated bacteria, for example, but they do really have a role even in protection against viral infections. And it has also been found that the risk of developing this poliovirus-induced infection after vaccination, it was actually lower in patients with severe combined immunodeficiency compared to those with X-linked agammaglobulinemia, for example, just highlighting again the role of the B-cell in protection against viral infections. It has been found during this pandemic that patients who have got common variable immunodeficiency have had a higher number of deaths compared to those that did not have common variable immunodeficiency, again highlighting the importance of the B-cell in protection against viral pathogens. B-cell depleting therapies like rituximab, I recall there's a child I've been treating for lupus, and she did not respond to, she has lupus nephritis, she did not respond to mycophenolate mofetil, and this happened at a time when we were in the second wave of the pandemic in my country. So the issue of admitting her for 6 monthly courses of cyclophosphamide wasn't very feasible because of fear of contracting COVID. So we opted actually to give her a dose of rituximab. And it's been 2-3 months since she got her dose and she developed herpes infection, that every time we withdraw valacyclovir, her herpes recurs, and she only received 1 dose. So, as we also institute some of these immunotherapies, let us be cognizant of some of the infections our patients are at risk of getting. And I've just highlighted some of the other viral infections that you could see with B-cell depleting therapies like rituximab or belimumab. What are the challenges we face? So here you are, you have a patient with history that you suspect is an inborn area of immunity. You have even narrowed down and you suspect that it's potentially a B-cell defect. And some of the challenges, and I was just sharing in some of the things I've experienced and from literature, is that viral infections can be very difficult to diagnose. The diagnostic tests are not necessarily widely available. And the serological methods that we use, when you're looking for antibodies against a particular pathogen are, often unreliable in our patients. And sometimes you're getting them and they've already received courses of immunoglobulin therapy. So that makes it even more difficult to make the diagnosis. And viral infections don't seem to be a very hot topic and key area of interest like the bacterial infections. And of course, because of the numbers of these patients, we do not have large data to be able to generalize. Thank you. So I don't know why my screen has frozen. Let me see. Almost done. All right. So to summarize, I cannot overemphasize the importance of history and clinical examination are key in recognizing potential clinical patterns that suggest B-cell defects. And that B-cell defense response is important not only for bacterial pathogens, but also viral pathogens. And a systematic clinical approach is really key for early recognition and prompt management to ultimately improve outcomes for our patients who truly deserve the best. I'll end with a quote by a Pan-Africanist, Kwame Nkrumah, who said, "It is clear that we must find an African solution to our problems, and that this can only be found in African unity. Divided we are weak. United Africa could become one of the greatest forces for good in the world." And in a special way, I really want to commend the African Society for Immunodeficiencies, because you have literally walked the talk. You've been able to bring us together. You continue to do so, and you inspire some of the other specialties like pediatric rheumatology. So a big thank you to our patients who inspire us to improve and be better healthcare providers. A special thank you to Dr. Nahla Erwa and Dr. Reem Elfeky for their guidance and support as I prepared for this Congress workshop. And ultimately, a big thank you to all my mentors and teachers in my professional journey. Thank you so much.
Hello and good morning from rainy Manchester. So thank you for having me today. My name is Shuayb. I'm one of the immunologists here in Manchester, United Kingdom. So thank you for asking me to come over for this amazing and exciting workshop. I can see already the awesome talks being done by Mirjam and Nahla and Angela. You've already kind of covered the whole B-cell defect. I think you haven't left much for us. So I'm trying basically today to go through the diagnostic of B-cell defect in kind of a holistic approach. Obviously, sometimes you do a whole lecture or a whole session or workshop for only diagnostics. So it will be the whole day. So try to condense this in half an hour sometimes will be a challenge, but obviously I'll try to give the highlight of what we're thinking about. And at the end of the talks, we're going to have a question and answer session. So please post your questions in the YouTube channel, and we'll try to select them going through the Q&A. So you already know, obviously, from the talks already being delivered before me, we dealing with the main immune deficiency is antibody deficiency, which is more than 50 or 60% of the primary immune deficiency. I would like to get your attention that, and you already know that, when we divide the immune system compartments into B cell defect and T cell defect and phagocytic defects or complement defects, we're only doing this from the academic perspective, so we can describe to the trainee how this work, and as well for us to understand how each compartments work. But at the end of the day, they all work together in a very beautiful teamwork, and you cannot ignore one without the other. So even when we talk about the pathogens, for example, like what Angela mentioned, in the B cell defect, you see a lot of overlap with other immune deficiency. So when we talk about B cell defect or humoral immunodeficiency, as you're already aware about this by now, we're talking about defect only within the B cell, either intrinsic to the B cell or interaction between the B cell and T cells or other lymphocyte. And the clinical presentations of these, when you've already seen from Nahla talks about the different presentation from early childhood, early as 6 months or even earlier than that sometimes, but 6 months is a common age we see, and then all the way down to adulthood. And sometimes this primary antibody deficiency can start for the first time in their 40s and 50s. So especially common verbal deficiency and similar antibody deficiency. The other important aspect, which already been described by Nahla and Angela, the comparison between primary and secondary. And I still struggle with this day and night when I have these patients coming to me in clinic. Are these primary or secondary, and how can we differentiate that based on the history, but also on the diagnostics we're going to do? There's plenty of classifications exist. And obviously you've seen from Mirjam talking about the development of B cell all the way down to the classification being beautifully described by Nahla from our IUIS classifications 2019. So I'm trying to put it in a simple way, just from the diagnostic perspectives. So try to divide them in defect in the early B cell development. So this the first lecture that Mirjam talked about, all these early stages of B cell, and all the defects occur in the early stage of B cell. Most probably will be in the bone marrow most of the time. And then you've got the second stage, which sometimes we describe them as hyper IgM syndrome, which again is a misnomer, but we still use that. And that's all the classes switch recombination defects. And that's within the B cell most of the time, but sometimes the interaction of B cells with T cell. And then I put CVID as a separate category because this is a complex immune deficiency with, we're still learning about all the presentations, phenotypes, diagnostics, and how can we monitor and follow up these patients. And then the other group included the secondary antibody deficiency, which is increasing dramatically. And I'm sure Angela would agree with me. All these medications they use in the rheumatology world causing a lot of problem in the immune system, but not only rheumatology, obviously gastroenterology, neurology, and all the other ologies with all these new maps and biologics from pure CD20 blocking agents all the way down to JAK inhibitor, they all affecting our immune system. So I've tried to put all these others, which include SPAD, specific antibody deficiency, selective IgA deficiency, and so on. You don't need, I mean, in a lecture like this for immunologists, you're already aware about the common acronym we use, the severe infections, persistent infections, unusual infections, and recurrent infections. So I don't need to go in detail with that. And obviously, Angela have described beautifully how this pattern of infections would target our investigations, because they don't want you to come into the clinic and say, I'm going to request like 20 tests for this patient to figure out the immune deficiency. We are here to learn about how we be cost effective in our request, and particularly in our countries in Africa, we try to be the most cost effective for a patient, especially if we're dealing in a private healthcare, what the easiest and best test we can do to get to the bottom of this medical problem or this immune deficiency. And Angela already described beautifully all different pathogens that can present and suspecting which immune deficiency. And obviously, I can't stress more. I mean, you've seen already, there's been stress from Mirjam to Nahla to Angela and myself today about the viruses in B-cell defect, and particularly with this pandemic, we've seen a lot of problems. Classical example, enterovirus encephalitis in children. Most probably these patients might have XLA, but obviously each defect will gonna be giving you some idea of which immune deficiency would be. The other important things to think about in immune deficiency, particularly we're talking about B-cell defect here, is not only the recurrent infections, there is as well overlap with other defects such as recurrent autoimmune or multiple autoimmune conditions, malignancies, and particularly with CVID, we've seen a very high risk in or increase in certain malignancies. The same apply to allergy and anti-inflammatory conditions, as well as metabolic disorder. So be aware about all the potential associations with these immune defects. The other important aspect to consider when we talking about the diagnostic of B cells is the age of the patient. When we're requesting lymphocyte subsets, for example, in a newborn, that would be different from somebody in the preschool or in adult or adolescent life. So that's really important. The difference range is the difference between that. And that's a lot of calls I get from various specialists about these reference ranges, and have to, I'm not going to say you're going to memorize that, but obviously put it in consideration. So one of the common start of the diagnostic age will start from the 6-month-old onwards. And this is the classical diseases we see from the B-cell defect, XLA, hyper IgM syndrome, or transient hypergamma of infancy. Again, some syndromic presentation can start at this age, which as well might have some antibody deficiency. Very simple lab test. If you've done just a full blood count and immunoglobulins, you can figure out we're actually dealing with antibody deficiency. School age, at the age of between 7 and 12, we're going to start seeing the emergence of the CVID, as well as the specific antibody deficiency, another syndromic defect like WHIM syndrome, when you have lots of warts and hypergammaglobulinemia. Again, one of the basic tests we can start with, just a full blood count, immunoglobulin levels. Specific antibodies, I'll come to that at the end of the session. But then other tests obviously will be important in the context of other suspected immune deficiency. But remember, always start with very basic tests, and that will give you the clue. But obviously, the key is you need to suspect that from the first place before we're going to say, you know, this is an antibody deficiency. When it comes to adult life, other CVID will be more common, and SPAD or specific antibody deficiency, we see that more appearing towards the adult life, and particularly in the context of other illnesses. So the two main goals when I deal with the diagnostic of a B-cell defect or any immunological assessment, I've got two main factors I need to think about. I need to think about the numbers and the function. And please remember, this is really important. All the tests we do is all about the numbers and the function. The number, either you do a flow cytometry or just a simple full blood count to give you the number. And the function, it's simply the function of B cell, and you can assess that by different ways, like vaccines to start with, but there's other ways we can assess the function of the B cell at kind of a whole compartment, or specific function of B cell proteins and cytokines. And remember here, as immunologists, you're supposed to be clever, obviously, and I'm sure you all appreciate that by now, going through the training of immunology. You're going to have to solve this puzzle with the age of the patient in one side, the history of the patient as already described by my previous speakers, and you're going to see more case presentation by Hiba and other presentation at the end of this workshop. Microorganisms, you've seen that beautifully by Angela, and that's really important. And then the affected organs, you try to put all the pieces together with the test you're going to do to come up with the final picture. So it's a quite clever work, and I think it's very satisfactory, especially when you manage to not only diagnose the patient, but also to treat the patient. And I always say this to my trainees: trust your gut instinct. And it is very simple. If it looks like a dog and walks like a dog and quacks like a dog and it smells like a dog, it's probably be a dog. So try to kind of have the common sense as well. You don't have to always do all the genetic testing in the world to come up with the diagnosis. So the whole test we're going to be talking about is numbers and function. So the numbers start from the full blood count. So that's really important. When you look into the total lymphocyte number, that will give you a general idea what the lymphocyte look like. And obviously, if there's lymphopenia, then you're going to do lymphocyte subsets. So you look for the TB and NK cells to check whether these cells in numbers, they are kind of okay or not. And obviously, if you've got somebody, say, for example, absent B cell, you're going to start thinking, okay, here now we have got a few things you need to think about. A gamma globulinemia is one of the things you're going to think about. Those patients have got no B cell, or could be secondary if they receive rituximab. So you're going to put that in your mind. Again, we've seen a lot of patients with good syndrome. So that thymoma with immunodeficiency, they have absent B cell. So you're going to think certain thoughts in your head come through when you're thinking about the absent B cell in the lymphocyte subsets. Obviously, when you're dealing with a female, then you're going to think about the other autosomal recessive disorder that may be associated with absent B cell. That's just to give you some examples. And then you go ahead and do the quantitative assessment for the immunoglobulin. So that would be your IgG, M, A, and E. And obviously, in that case, you look for the numbers of these antibodies. So here, only the numbers you've been assessing so far, your lymphocytes and your immunoglobulin. Then you can proceed to the function. And when you come into the function, I mean, by that time, you already have some idea what we're dealing with from the perspective of immune deficiency. Now, when you come to the function, one of the main tests we do in the clinic is a vaccine titer. And obviously, every country has got their own guidelines for this. And obviously, all the examples I'm going to give you is only based on our experience here in the United Kingdom. So the vaccine we use here for B cell defect mainly start with a polysaccharide simple pneumovax or salmonella vaccine. And obviously, it depends on the area where you suspect these patients might have more vaccines. So for example, here, I can use salmonella typhi polysaccharide or pneumovax, or the polysaccharide pneumococcal vaccine. But in Sudan or Africa, I wouldn't use salmonella because you've got plenty of salmonella there. So we're going to be most probably using pneumococcal polysaccharide vaccine as a first step. If the patient already received that, or you haven't seen a response following the vaccine, then you may think about Prevnar 13, which is a conjugated vaccine. There's plenty of conjugated vaccine we use. One of the examples is Prevnar 13, which is a conjugated pneumococcal vaccine, but also Haemophilus, tetanus, and a lot of other vaccine can be used as a conjugated vaccine. And that will tell the relationship of B cell and T cell. So when you test a polysaccharide on its own, you're testing the function of the B cell on its own. When you're testing the conjugated product or conjugated vaccine, then in that case, you're testing the relationship between B cell and T cell. And obviously, if the relationship is still intact, then they should produce a good response. If there's any problem within this relationship or the B cell have got defect, then you're not going to see a good response. And the test goes on with the function of the B cells and so on. One of the challenging questions we have at the moment, particularly when you have really advances in immune deficiency diagnostic, particularly in the last 3-4 years, we had expansion in the facilities and in the diagnostic tools of primary and secondary immune deficiency. We used to have only flow cytometry is just the main test we do, but obviously we're talking about functional assays and we're talking about genetic testing. And many centers at the moment in Africa and as well in other countries, particularly here in the United Kingdom and the West, we've got a lot of genetic testing going to take place. So that's why it became the approach in 2021 for a lot of patients. Obviously, we still do the numbers, we still do the functional assays, but there's a lot of genetic testing taking place. That's particularly in other immune deficiencies in complement, T cell defect and phagocytic defect. In the B cell defect, you get the diagnosis from the first test you do. But particular patients, you may need to do genetics, particularly if you've got a young female at the reproductive age, and she's got a gamma globulinemia. You would like to know what's happening there. That's one of the examples. But genetics has been expanding dramatically in the last few years. You don't know, you don't want a definition for XLA as in an immunology workshop, and you know by now how common this defect is. In the northwest of England here, we look after 10 patients with XLA, and I think we most probably have in everyone we need to have with regards to XLA. The main issue we see with XLA from the diagnostic perspective and management perspective, in the past, we say, give them the IgG, you sorted, you have got no problem. But now we see a lot of other complications we never thought about, because remember, this XLA patient have got no B cell, have got none of the immunoglobulins, no IgG A or M. So by the time they come to adulthood, they have got no IgA at all in their bowels, and a lot of these patients start developing inflammatory component to their bowel, and we've been seeing a lot of inflammatory bowel disease, atypical, not typical of Crohn's or ulcerative colitis, some sort of a different inflammation. But also we've seen some blood disorders in these patients, and we start seeing more and more. And I think we only, as we always say, we learn from our patients how we treat this condition. When it comes to the diagnostics, these XLA, they're not going to have any B cells as you expect. T cells might be normal. They're not going to have any immunoglobulins at all. I'm just going to spend a few minutes on CREX. So CREX is kind of one of the new tools now we've been talking about in the newborn screening, and some countries already started that. So I'm going to spend a few seconds in CREX. So when we come to the diagnostic from the flow cytometry, already Nahla alluded to that. So if you, for example, have a normal person here, a control, XLA patient, and a carrier, you've got here in the y-axis, you see the 19 here and see the 14 here, and in the x-axis, we've got the BTK expression. So in your XLA patient, you're not going to find any BTK expression in a classical XLA. However, in the carrier, you're going to see two population, and that's classically in the CD14, which is a monocyte. You're going to see a proper classical 2 population. So this is really simple, very straightforward. If you have got the test at hand, that's a diagnostic. You don't really need the genetic testing for that, and you can just go ahead and treat this patient as BTK defect or XLA in the patient. Now let's spend a bit in the CREC cycle. You've already seen the development of the B cell from the brilliant talk of Mirjam in the first session this morning, all about the B cell defect and all that. If you remember when Mirjam talked about the class switch and all these recombination circle, we've got a few if you like, bystanders or some product of this complicated and clever pathway. And one of the classical product we see, we call it CREX. So that's the kappa recombination. Sometimes we call it excision circles or excision, mainly excision circles. That's kind of the common talk we talk about. So these CREX is basically little circles in the naive B cell, and the number of the CREX reflects the number of your naive B cell. The same we see in the T cell, we call them TREX, so T receptor or T recombination excision circle. And obviously these circles is literally, if you have got a lot of them, then you most probably have enough naive B or T cells, and that will reflect most probably they have a normal B or T cell. Obviously not always the case, because you can have a proper normal number of B or naive B cell, but you've got the defect somewhere else. Now, how do we do this test? In certain countries now, as a newborn screening, we've got this Goth record. I'm sure you've seen that, in many neonatal units when they prick the heel of the newborn and get some drops on this Guthrie card to analyze for certain inborn errors of for example, metabolisms or other inborn errors of certain severe illnesses. We try now to introduce this inborn errors of immunity to the newborn screening. There's a lot of countries that started that in the world. America is obviously gone a long way with this. Europe have done, again, massive work, and I'm sure Mirjam would agree with me, particularly Netherlands and other countries over there in Europe, they've done a massive work, and they publish a lot of work on that. In the UK, we just started the trial. We're always behind sometimes in Europe because we need to make sure we're really dealing with the right thing. I don't know. But anyway, we started the trial only this September for the first time to actually roll it out for the whole newborns in the United Kingdoms. That's going to be a trial for the next year or so. And then following that, hopefully will be part of the normal screening we're going to offer for all our newborns. And I'm hoping in Africa, we're starting this, or we already started this. And I'm sure the other expert in Africa can tell me how far we are with this newborn screening. Gamma globulinemia, this is other causes of similar presentation to XLA, but you see this particularly in females, but obviously in other, it could as well be in males. But again, the list is, this is just a very small list of autosomal recessive we see in those patients, but the list goes on and on. Every single day, we get a new genetic defect. Yesterday, I had a new report for one of my patients with a gamma globulinemia. That's totally never expected. So that's kind of all about a gamma globulinemia. Few words about hyper-IgM. Again, this is a very old name, but I still will love to use it. And as a trainee, try to use a new classification as a class switch recombination defects. The common four, which we always talk about, your X-linked hyper-IgM syndrome, and then you've got your autosomal recessive disorders. We're going to start from CD40 deficiency to activation-induced cytidine deaminase defect to uracil and glycosylase deficiency. Now, these patients obviously can present with various defects, and you've seen that beautifully in the previous talks. From the diagnostic perspective, you're going to look into the B cell defect. Again, the clinical phenotype is quite variable, and that depends on where the defects are. But generally speaking, if you've got a defect in the relationship or the communication between B cell and T cell, we most probably going to see a combined immune deficiency picture. If the defect only within the B cell, then you're going to see only B cell defect phenotype, and that would be mainly, as we mentioned earlier, with the pattern of infection, would be mainly bacterial infections and certain viral infections. So the B cells versus T cells is quite important. In the immunoglobulin levels, you're going to see mainly normal or high levels of IgM, and sometimes we see a bit low other immunoglobulins, and this is again quite variable. It depends on what defects, and the defects are quite complex sometimes. Vaccine responses, again, as we mentioned earlier in the beginning, that depends on where the defects, and if you've got defect only within the B cell, then you might have a failure to respond to a polysaccharide vaccine. But then if the defects in the combined way or in the CD40 ligand, for example, then those patients might fail to respond to both polysaccharide and conjugated vaccine. And classically, when you're going to see this a lot in my slides, when you see the phenotypes of the B cell, starting from naive B cell on the far right lower corner here up to anti-switch memory to class switch memory B cell, and that's where the defect will be in this hyper IgM syndrome. Classically, in a phenotype, in a flow cytometry picture, for example, here if you have got the cells and you've got here the CD40 ligand in an X-linked hyper IgM syndrome, you're not going to have any expression of the CD40 ligand, but in the carrier, you're going to have two hubs or two population, and that's a classical example. And I mean, you already agree with me. These investigations in the lab is quite straightforward if you have got the right technology, and it's all about the visual presentation of such technology. The list of hyper IgM syndrome or class switch recombination defects, it's pretty long, and we keep seeing more and more defects. And as already mentioned by Nahla and Angela about the pattern of recognition, these patients sometimes will be syndromic. So you look into other kind of either their faces, their skin, their other problems that will help you with the diagnostic. I had a patient with hyper IgM syndrome, a young lady, and I could not figure out what the genetic defect for her. We've done so many immune panels for her, but finally 3 weeks ago, we've just received a very rare mutation in the ICOS gene. So these are sometimes very challenging patients, but we know they have got defect in this compartments and in the recombination defect. We're going to give them the treatment, but obviously if we know the genetic defect, sometimes will be easier, particularly if we're thinking about transplant. And again, transplant is not always possible in such patient. Thank you. We'll try to concentrate on the last few minutes about CVID and just a few words about others. CVID is the commonest, by far, the commonest primary immune deficiency we see in adult, and I'm an adult immunologist, so I see a lot of that in my clinic. And you can see here, they are variable, so they can have multiple faces, and we see different presentations of these all the time. I classify them as an umbrella of immune deficiency, because within that umbrella, you can see a lot of defect. This is now a relatively old publication from 2016, but reflecting all these monogenetics we see is roughly 10% of all the CVID patient. So in my center here, we have got more than 160 patient diagnosed with CVID, and the genetic or monogenetic diagnosis, I don't have more than 20 patients or so. So that's this percentage of 10% is always right. In the phenotype, the most important things we need to think about is, and I'm sure you've seen this in the publication, the famous publication from Euroclass about the different phenotypes of B cell. And the main one you need to concentrate about are those three. So we're talking about the class switch memory B cell, which we talked about early, and then you've got your activated B cell, and that's your CD21 low, and you've got the transitional B cell. You want these activated and transition to be low, and your class switch to be high. If those be high, if the activated B cell has been high, this can link to a lot of complication, particularly splenomegaly and all that, and the similar to transitional B cell, that can link to a lot of lymphadenopathy. Classical flow cytometry phenotype for these cells, a very simple representative will be this picture of your IgD in one side and CD27 in the x-axis. The far lower here, you've got the naive, and going all the way down here, will be your switch B cell. Remember always, the class switch memory B cell is the most important one, and this is really practically I've seen in my center how the low class switch memory B cell may link to very devastating complications, particularly this granulomatous complication is growing more and more. If you ask me 10 years ago, I would say, yeah, we know there's some granulomatous complication in this CVID patient, but we don't really see that much. But nowadays, we see a lot of that. I'm just going to show you one of the examples in a few seconds. The agreed, I mean, we use it here, obviously, in the United Kingdom, and I'm sure in other countries, they may have some different cutoff, but more than 90% of transitional B cell, we've seen a lot of lymphadenopathy in these patients, and sometimes we even need some immune suppression to control that, and we've seen the same with CD21, particularly here in Manchester. This is just one of the example of one of my patients in 2015. You can see how beautiful she looks, and obviously she's still beautiful, but in 2017, bless her, she almost lost her nose because of the granulomatous complication. She started having some nasal speech in 2015, and by 2016, she had a massive hole in her hard palate, and we had to do constructive surgery to control that. Her granulomatous complication literally eating her from all over, from inside, from her bowel, from her skin, from her nose. And we had to rush to a transplant. Unfortunately, she couldn't make it. And the transplant was only a few months ago because we just struggled with COVID and all that. But that's just one of the examples. Those patients with granulomatous complication can have very devastating outcome. To finish with, in the last few minutes, we'll just talk about this very briefly. Secondary immune deficiency is growing really fast. You need to look at that all the time. Simple test, full blood count, and immunoglobulin will give you the answer. Selective IgA deficiency is quite common in the UK here and in the Caucasian population, almost 1 in 500. I have no idea how common in Africa. I'm sure the expert in Africa might tell us how common it is. Generally speaking, I don't see much problem because it's quite common, though we have got few cohort of them may present with celiac disease and other bowel problems, or even inflammatory bowel disease and other sinopulmonary infections. SPAD, or specific polysaccharide antibody deficiency, again, is kind of increasing population. Most of them, they kind of manage okay with antibiotic prophylaxis. Very few of them require immunoglobulin replacement therapy. Selective IgM, again, this is a defect. Sometimes we see in IgM deficiency. To be honest, it's quite rare. Haven't seen any patient with that, only for IgM, but we see a lot of low IgM in secondary immune deficiency, and again, not necessarily linked to a problem. And obviously, I've got the transient hypogammaglobulinemia of infancy, and that's only a transient, as the name imply. In the secondary, the main thing I would like you to remember is where the defect is. It only humoral, or is it cellular when you do the full blood count, or is it a mix together? Always remember, is it a primary or secondary? And that's based on your history, and the diagnostic will be the answer. The drug, which drug, and other causes has been excluded. Remember your basal, you're going to have to join them together. And again, sometimes the actual underlying illness could be a problem. And we've seen that in, so for example, SLE patient, we do badly with immunosuppression compared to other rheumatological disorder. So that's really important in your diagnostic tool. So in summary, the clinical phenotype is the key to direct you how you're going to order your diagnostic. You're going to start with a very simple laboratory immunophenotyping of B cell, based flow cytometry and numbers of B cell. Then we can talk about the B cell functions. And obviously here you might actually reach the final diagnosis when we talk about the B cell defect. Very rarely we need some genetics. And obviously there's a lot of literature you can have a look to read. The final things I usually like to finish with is this well-known set from Dr. Robert Goods, the doctor who described Goods syndrome, always say the greatest teacher of modern immunology, patients with immune deficiency disease. I would like to thank you all, guys, and we'll share these slides, I'm sure, in somewhere and how, and you've got a lot of reference, you can have a look on them. Thank you ever so much.
Hello, I'm Hiba Shendi. I'm a Sudanese immunologist working in the United Arab Emirates. I would like to thank the organizers for asking me to speak today and thank my colleagues who have given excellent talks on B-cell defects. I will be presenting a few clinical cases, all relevant to the talks that have been previously discussed. So my first patient is now an 11-year-old boy who's been troubled with recurrent chest, ear infections, and recurrent diarrhea, started at the age of 6 months. He received the rotavirus vaccine at around 2 months and got gastroenteritis after that. At 2 years, he had bacterial pneumonia complicated with empyema, and that's when the pediatrician thought about immunodeficiency. After that, he continued to have a chronic cough and also has problems with conjunctivitis. He's failing to thrive. Family history, parents are related. He has an older brother who's healthy and well. Examination revealed a small child with height and weight below the 3rd centile, and the other remaining examination was unremarkable apart from absent tonsils. So because of the current infections and being a male, he had his immunoglobulins checked. These show undetectable IgG and IgA and a very low IgM. The low IgG subclasses do reflect the very low total IgG level. So from previous speakers, the next step would be to look at his CBC. It's always useful, and that showed a microscopic hypochromic anemia. Whenever I see a result, I always like to confirm it, especially when it's very unusual. And this showed that he does have a significant hypogammaglobulinemia. So we proceeded to lymphocyte subset analysis. And if you look at the results, you'd be able to see that he has high CD3 levels, so high T cells with high CD4 and high CD8. And if you look at his CD19 B cells, you'd find that they're very, very, very low, almost undetectable. So the normal is 400 and he's got about 5. So from the clinical presentation, being a male presenting with recurrent sinopulmonary infections, diarrhea, having absent tonsils, and having hypogammaglobulinemia in the presence of absent B cells, that would really direct you to X-linked agammaglobulinemia as the diagnosis. So that's enough diagnosis for us to manage him. He was started on immunoglobulin replacement therapy. Because of his ongoing chest problems and cough, he had a CT scan which showed that he had lower lobe bronchiectasis. He was also commenced on prophylactic erythromycin. He was advised not to receive any live vaccines, and actually no vaccines except annual flu vaccine, because he was on immunoglobulin replacement therapy and his response would be incomplete. His acute infection is much better. He still gets a cough now and then, and conjunctivitis is an ongoing problem with him. He's actually now on regular prophylactic topical antimicrobials to control this. His weight has increased, but he still remains below the 3rd centile, and we're increasing his IgG dose further now. He's almost on 1 gram per kg, hoping to improve his chronic cough and hoping to see better weight increase. So other investigations to think about, anything, I mean, I think we're pretty confident. We're not 100% sure, but we're pretty confident it's XLA. Having consanguinity in the family, he may have autosomal recessive form, which really would make you think, as actually it was the parents' question, what about other children? What do we do next? And that's when, if you can do genetic testing, it can be helpful. He actually does have a mutation in the Bruton tyrosine kinase, so it's confirmed XLA. And the genetic counseling is because they're planning to have other children and they're considering pre-implantation diagnosis, but not for therapy. So what's Bruton's agammaglobulinemia? It's a defective maturation of B cells due to Bruton tyrosine kinase deficiency. Typically, they present early in infancy with recurrent sinopulmonary infections, parasitic infections like Giardia, They can get cellulitis, abscesses, they can get diarrhea, and enteroviral viruses are a big problem. That, as Angela had mentioned, oral polio vaccine is one thing that you need to avoid with these children. On examination, they may have absent or very, very small tonsils, and you'd find that they have very, very low immunoglobulins with absent or very low B cells, and the mainstay of therapy is immunoglobulin replacement therapy unless they develop other features other than infections. So our second patient is a 17-year-old female. She presented to immunology at the age of 11 years, and at that time she had a history of recurrent chest infections and recurrent otitis media, which was quite severe. That has been happening for about 6 years. She also had a chronic productive cough and has history of chronic diarrhea. Actually, the referral to immunology was after this biopsy result that showed that she had local villus atrophy and intraepithelial lymphocytosis with absent plasma cells. And it's actually the histopathologist here who raised the possibility of an immune deficiency because of the lack of plasma cells in the biopsy. She was also short and small for age. Her family history was parents were not related, and she had siblings who were well and healthy. On examination, she had clubbing. The tonsils were there, but they're not large, and she had some chest crepitations when we saw her in clinic. So because of the history, again, of recurrent sinopulmonary infections, having a chronic diarrhea, which makes us suspicious of either a gut infection or an autoimmune enteropathy, and with these features, it went more with an autoimmune enteropathy with absent plasma cells. She went for immunological investigations. As we always say, don't forget your TBC, look at your neutrophils, look at your lymphocytes, and immunoglobulins. And she has a pan-hypogammaglobulinemia that was confirmed. So she has very, very low immunoglobulins. So we proceeded to check her lymphocyte subset. And if you look at them, she's got normal T cell numbers, normal CD4 numbers, CD8 numbers, and normal B cells. So she completely normal lymphocyte subset analysis. So what did we do next? Okay, we needed, for her clinical picture, we needed to do a CT scan. She had stools, first stools for infections. There was no evidence of infection. Unfortunately, due to cost limitations, and we didn't get an HRCT scan. We always need to look for secondary causes of hypogammaglobulinemia. She had normal albumin, she had no protein in urine. From the history, she wasn't on any drugs like chemotherapy that would cause low immunoglobulins. In an older patient, in an adult Hello. Sorry, everybody, for the technical issue. We'll just carry on where we stopped. So yes, we need to do an HRCT scan because of her ongoing cough and clubbing, but we were unable to do that because of cost limitations. We need to exclude a secondary immunodeficiency. So we looked at, she had normal protein, she had no proteinuria, normal liver and kidney function test. In an older patient, if the history is suggestive, you need to look for lymphoproliferative disease by an HRCT or a CT scan of the chest, abdomen, and pelvis, or consider looking for urine for Bence-Jones protein, and check if they're on any drugs that can cause immunosuppression, which he's not on. So so this is a patient with a significant history of recurrent sinopulmonary infections, chronic diarrhea that with evidence of autoimmune nephropathy, and low immunoglobulins but normal lymphocyte subsets. So we proceeded to do vaccination studies, and she was vaccinated with pneumovax and tetanus. If you look at her post-vaccination responses to pneumovax, they were completely flat, no response whatsoever. And her responses to tetanus were normal. So based on that, she was diagnosed with common variable immunodeficiency. And looking at what criteria she's meeting, first we've got low IgG, A and M. Of the second group, she's got increased susceptibility to infection and autoimmune nephropathy. She does not have granulomatous disease, no lymphoproliferation, and no family history. We showed that she does not respond to polysaccharide vaccines. We don't have the facility to measure memory B cells, and we were able to exclude secondary causes of immune deficiency, and she's over 4 years of age. It's also important to exclude combined or less profound combined immunodeficiencies and make sure the T cell compartment is not affected. Clinically, she does not have fungal infections, she does not have viral infections or opportunistic infections, such as the T cell defect. So she was started on IVIG replacement therapy, and her response was magnificent, really. Her weight just went up, really, really well. She was also, because of her clinical findings that are suggestive of bronchiectasis, although at that time didn't have a CT, we then had it, and she does have bronchiectasis. And she's also being managed with a pulmonologist. Her diarrhea also improved significantly on immunoglobulin replacement therapy. Our third case is that of a 2-year-old female who's full term. She presented at 6 months to the immunology clinic. Her parents are cousins, and she had one older sibling who was diagnosed with disseminated tuberculosis after the BCG vaccine. He was failing to thrive, and at that time he was investigated and found to have a T negative, B negative, NK positive skin. Unfortunately, he was not transplanted early, and he passed away. So she was referred to the immunology clinic for evaluation to make sure that she didn't have SCID. At the time, she was growing well, she was thriving, she was breastfeeding, she had no infections whatsoever, no thrush, no skin rashes, and she had not received, luckily, any vaccines. Her examination was completely normal, and her tonsils were present. So because of the history, family history of SCID, we went on and we checked her immunoglobulins, and we found that her IgG was reduced, as well as her IgA. IgM was normal. We proceeded to check lymphocyte subsets urgently, really, because of the family history. And we were very relieved to find that she had normal CD3 cells, normal CD4, CD8 cells, as well as B cells. So she doesn't have the same results as her brothers had. Can we exclude SCID? It becomes less likely, but we can't completely exclude it because we know that there are atypical forms of SCID. Her CBC was also entirely normal. It would have been indicated at that stage to look for TREX or naive CD4 cells or lymphocyte proliferation, and unfortunately, we did not have any of that available. We usually could send for TREX, but that's a send-away test, and with the COVID situation, the samples, we could not get the samples to the lab on time. Having a molecular test would have helped. Having a molecular result on her brother, we could have tested her for that specific gene mutation, but again, we didn't have a result on the brother. So at that point, all we could do was actually just monitor the patient, watch her really, really closely. We were significantly reassured by the lymphocyte subset results, and we had their very close. And the parents were aware if she gets any infections that she needs to come to our attention as soon as possible. So we continued to monitor her and monitor her immunoglobulins. And as you can see, over an 18-month period, by the time she was 2, her IgG and IgA completely normalized. She remains very well, no infections, and she started her vaccination now. So we diagnosed her with transient hypogammaglobulinemia of infancy, which is a prolongation of the physiological nadir that usually would see at around between the age 6 and 9 months when infants lose their maternal IgG and take a bit of time to start making their own. This can be prolonged up until 4 years of age, and it's more exaggerated and more prominent in premature babies. Usually IgG is reduced, but we can see reduced IgA, same as our patients. And most patients, same as our patients, do not have any infections. And a minority do. And as a physician, you're always guided by the clinical history. And if they do have significant infections or recurrent or troublesome infections, you may consider a transient course of prophylactic antimicrobials or immunoglobulin replacement therapy while monitoring them until they reach normality. And it's a retrospective diagnosis because you can't make it until the immunoglobulin is normalized. So the fourth case is a 4-year-old female who was diagnosed with insulin-dependent diabetes mellitus at the age of 3. She continues on insulin. So somebody was screening her for celiac disease, I think, because of the association of insulin-dependent diabetes mellitus and celiac disease, and she was found to have undetectable IgA levels. She does not have any diarrhea or symptoms that suggest celiac disease. She does not have any infections, no chest, no ear infections, no thrush, no pneumonias, no abscesses, very well apart from her diabetes. No recent antibiotics. She does not have any allergies, eczema, asthma, and she's a thriving child. So when we look at her test, her CBC is normal as well. Her IgG was slightly elevated, just above the reference for normal, and that's reflected in a slightly elevated IgG2. And her IgA is undetectable, less than 0.005, and her IgM is normal. We did her lymphocyte subsets, which again were not really significant, very borderline reduction in CD3, CD4, and CD19 levels. She had her normal vaccination, routine vaccines, which would have included Prevnar, tetanus, and Haemophilus, and we could see 7 out of 10 pneumococcal serotypes were greater than 1.3, and 11 and 10 out of 12 were greater than 0.35. So we were happy with her responses with pneumococcal and tetanus. And being clinically very, very well, the diagnosis was selective IgA deficiency, which is defined as an IgA of less than 0.07 grams per liter in the presence of normal IgG and IgM. The child has to be greater than 4 years because we know that levels can normalize up to 4 years of age, that there are no other causes of hypogammaglobulinemia, and there are normal responses to vaccines. Most patients are asymptomatic, and there's a huge argument whether IgA deficiency or selective IgA deficiency is it a true immunodeficiency, and I think it's the symptomatic ones that we would regard as immunodeficient. Patients may have an increased risk of upper respiratory infections, allergies, and autoimmune disease, and it actually can be the first presentation of a more significant immune deficiency like CVID. My fifth and final case is a 29-year-old gentleman whose parents are related. He has healthy female siblings. He's long history of recurrent chest infection since the age of 2, when he was admitted for IV antibiotics for pneumonia. And after that, he continued to have recurrent chest infections as well as ear infections. He had grommets inserted in childhood. And at the age of 8, he was diagnosed with a left-sided bronchiectasis and had a lower lobe lobectomy. That was abroad in a different country. He was diagnosed with IgA deficiency, he told me in clinic, and was started on IVIG for 2 years and symptoms improved. He then moved countries and immunoglobulin replacement therapy was stopped. When I saw him in clinic, and that was quite recently, less than a year ago, he still was having 4 to 5 courses of oral antibiotics per year, mainly for chest and sinus infections. He had no thrush, no viral infections, no adenitis. Sometimes intermittently he would have loose stools, but no chronic diarrhea or chronic cough. He had a recent HRCT of his chest, and that did show bronchostatic changes. He was seeing a pulmonologist and was on serotype inhaler. His other problem, main problem, was that he had the panuveitis, for which he was following with ophthalmology, and that's responsive to topical steroids. So when I investigated him in clinic, could see that his IgG level was normal, IgA is undetectable, and his IgM level is low. IgG subclasses shows absent IgG4 and low IgG2 levels. Lymphocyte subset analysis was essentially normal, apart from mild increase in CD19 cells. He has a normal kappa to lambda ratio. And we proceeded to vaccine responses. Here we have a gentleman who has significant history of recurrent sinopulmonary infections, developed bronchiectasis, and has a pan-uveitis, who has IgA and IgG2 and IgG4 subclass deficiency. So definitely you could see the difference with him and with the girl with IgA deficiency who's completely asymptomatic. So he would not have received Prevnar in childhood at his age. And these are his pre-vaccinations, so that's the pneumococcal, 12 pneumococcal serotype, Haemophilus antibodies, and tetanus toxoid. Following that, he received Pneumovax, which is a polysaccharide vaccine, Haemophilus and tetanus vaccines, which are conjugate vaccines, so they have a protein antigen. So for pneumococcal vaccines, we would be assessing the pure B cell responses, whereas with the Haemophilus and tetanus, we'd be assessing both the T and B cell responses. And post-vaccination, 4 weeks after the vaccine, this is what we got: a completely flat response to pneumococcal, Pneumovax, so no response whatsoever, and a protective levels of Haemophilus influenzae and tetanus toxoid, which meets the criteria for specific antibody deficiency. So the diagnosis in this gentleman, he's got symptomatic IgA deficiency with IgG2, IgG4 deficiency, and a specific antibody deficiency. And I'm sure, if we can do switch memory B cells, we will find, the B cell subtypes, we will find some abnormalities there, and we might find a genetic diagnosis of one of the small genes that Nahla was talking about in the classification. And I think just to highlight the difference between case 4 and case 5, a completely asymptomatic IgA deficiency patient and an IgA deficiency with significant infections, bronchiectasis, and autoimmune disease. This is the one we need to pursue here further. This is the one we need to worry about. We need to treat and we need to monitor. So with that patient, with the last patient, he's on immunoglobulin replacement therapy now. He's on azithromycin prophylaxis. He continues to see ophthalmology who are considering monoclonal antibodies for his eye disease, and we referred him to gastroenterology to evaluate his intermittent loose stools. We're not in the position to do genetics now because of cost. So thank you very much for your attention. I'm happy to take questions later.
Thank you. Good morning, everyone. Apologies for my voice. I just have some flu, no COVID. And I'm going today to discuss management approach in patients with B-cell defect. My name is Reem Elfeky. I'm a pediatric immunologist and bone marrow transplant specialist who was trained in Egypt and currently working in Bristol Children's Hospitals. So I was going to recap on the B-cell defect, but I think all my colleagues have already discussed B-cell defects now, starting from Mirjam until Hiba. So I'm not going to discuss this. I will point to management for specific B-cell defects, management of associated complications and vaccinations in patients with B-cell defects. So I will start first with a collective approach of how we manage patients with infections who have an antibody defect. So we get infection history, we have full blood count, immunoglobulins, and we check lymphocyte subsets, and we divide the patients broadly to a gamma globulinemia, which is X-linked or autosomal recessive, common variable immune deficiency, hyper IgM syndrome, or a milder phenotype, which is selective IgA, polysaccharide antibody deficiency, or possible secondary antibody deficiency. And in terms of infections, Angela have beautifully described the infections that are common with patients with antibody deficiency. I'll just recap on this, saying for the sake of management. So patients with antibody deficiency, they have particular problem with handling extracellular bacteria, streptoneumia, Haemophilus influenza, and if they develop bronchiectasis, they are at risk of having Pseudomonas infection. They are also having problem with mucosal immunity, so it's not uncommon to find the Campylobacter infection, Giardia infection in these patients. So if we have a patient with selective IgA deficiency, polysaccharide defect, or secondary antibody deficiency, the first line of management is usually antibiotic prophylaxis. We usually give azithromycin, 10 milligram per kg per child, 3 times Monday, Wednesday, and Friday, and continue like this through the winter season. And if he's getting more frequent infection, it can be winter and summer. And if the patient is getting more than 3 significant infections per year, or if he develops bronchiectasis, or he is showing poor vaccine responses, then in this particular cohort of patients, we can consider immunoglobulin replacement therapy, continue antibiotic prophylaxis on top, and continue follow-up. In the more severe forms of B-cell defects, like agammaglobulinemia, X-linked or autosomal recessive, CVID, hyper IgM syndrome, we start with immunoglobulin replacement therapy. And the trough level of IgG that we keep the patient on is variable across centers. Back home, we were keeping them IgG around 5 and 6. Here in the UK, we were keeping IgG above 7 and X-linked agammaglobulinemia at 10, and I will discuss this later. And we'll monitor the patients. If they develop bronchiectasis, we need to optimize immunoglobulin levels and keep an IgG trough level between 8 and 12, and possibly add antibiotic prophylaxis as well. And we do a clinical evaluation, so assessment in clinic every 3-6 months. So we do pulmonary function test and CT scans every 3-5 years for unrevealing occult bronchiectasis. It can be, if the patients have bronchiectasis, maybe we need to do it more frequent. Lung function test, again, if there is evolving bronchiectasis or chronic lung disease, we might be doing it every 3 to 6 months. To monitor response to treatment. There are specific pathogens that in patients with antibody deficiency, as my colleagues have highlighted before. So in X-linked agammaglobulinemia, there is a problem with handling enteroviral infection, and I will discuss this in details afterwards. And the treatment so far that we know about is immunoglobulin IVIG, and the prophylaxis is again keeping the patients on immunoglobulin therapy. But as I said, I will discuss this in more details now. In patients with hyper IgM, and I'm just going to discuss CD40 ligand, CD40, just shedding some lights on them. So they are susceptible because there is a problem in the talk between the T and B cells. So these patients have problems in handling Cryptosporidium parvum, and we need to check the stools for this. We need to, if Cryptosporidium is there, because it can induce sclerosing cholangitis, we need to start nitazoxanide therapy, azithromycin, paromomycin, according to the where you are exactly. And prophylaxis, patients with this disease should have boiled water. Some centers can give some prophylaxis like azithromycin as well. They are also susceptible to another pathogen, which is the PCP. And again, treatment, if there is PCP pneumonitis, is high-dose cotrimoxazole plus short course of steroids. And for prophylaxis, we give septrin prophylaxis or IV nebulized pentamidine or dapsone if the patients cannot handle the septrin and have neutropenia and thrombocytopenia. So from here, I will just focus my talk on 3 different forms of immune deficiency that are linked to B-cell defects. So the first one of them is the most common one that we encounter, which is selective IgA deficiency. And beyond infections, these patients can develop other problems like autoimmunity. So 5 to 30% of the patients with the selective IgA can have autoimmune disease. They are at increased risk of celiac disease. If you see the picture on the right, 10 to 15 times risk than normal population to develop celiac disease, having abdominal bloating, nausea, vomiting, headache. And in these patients, you suspect celiac, but you are looking at IgA tissue transglutaminase, which will be negative because they have already no IgA. So what you should be doing is to look at IgG tissue transglutaminase. And of course, you can do endoscopy and see the atrophy of the villus atrophy and do a challenge with gluten, and then another colonoscopy or completely remove gluten from the diet and look at the response to gluten-free diet. They are also susceptible to other autoimmune disease, myasthenia gravis, ITB, autoimmune hemolytic anemia. There are some cases with autoimmune nephropathy and few patients with systemic lupus and rheumatoid arthritis. Why they are developed autoimmune disease? So IgA is quite important for mucosal immunity to keep like a first defense for the mucosa. So maybe there is an exposure to environmental antigens due to the absence of this IgA. And with molecular mimicry, there are cross-reaction with self-antigen that might cause autoreactive antibodies. In patients with selective IgA, we know that they have abnormal T regulatory cells. So it's not just a B cell defect. There is other cells that are also affected. There is genetic susceptibility, like HLA-B8, HLA-DQ2, for example, is more linked with the development of celiac disease and is present in patients with selective IgA deficiency. Saying this, there are patients that you can check and find that they have antibodies against thyroglobulin or red cells or basement membrane in the kidney, and they don't have any autoimmunity, and not necessarily that they will develop autoimmunity. So there is no preemptive treatment if you screen and find these antibodies. Other problem that these patients can develop is allergy. So 50% can develop allergy, possible cause, we say maybe compensatory raised IgE. Again, compromised mucosal barrier and possible absorption of allergens. So they can develop any form of this types of allergy, rhinitis, conjunctivitis, food allergy. And they can have reactions also to immunoglobulins and blood transfusion with anti-IgA antibodies that are IgG or IgE antibodies. And thus, in these patients, we can, if they require a blood transfusion for any reason, and we know that they develop the reaction, we can do washing of the red cells. There have been some people doing desensitization approach. If they are to start on immunoglobulin, it's better to use a low dose, a product that have low immunoglobulin A, and I just put some of these products here, or start from the beginning on subcutaneous immunoglobulin where you are not going to develop anaphylaxis or anaphylactoid reaction. Malignancy is, there is no clear increased risk of malignancy in patients with selective IgA, but again, they have a problem in the mucosal barrier and they are at risk of having H. pylori infection. And H. pylori has been linked in patients with primary immunodeficiency to the development of stomach cancer. As an evolution of how the disease will go, so 80%, around 80% of the patients are asymptomatic or they have minor symptoms, infection related. Few can completely revert to normal, and there is a potential for some patients to evolve into common variable immune deficiency. So if you have a patient with selective IgA deficiency, you need to have regular observations every 6 months, evaluation in clinic, trial of prophylactic antibiotic, as I said, winter season or all through the year, whatever, according to how much he's getting infections. Potential need for immunoglobulin replacement therapy, but be cautious, and management of allergy or autoimmunity as per usual guidance with the specialist teams. And then from here, going to the second thing, which is X-linked agammaglobulinemia. So I just want to say that actually X-linked agammaglobulinemia, this BTK is not only about B cells. So BTK is expressed in all hematological cells apart from the T cells. So BTK deficient neutrophils, for example, they undergo apoptosis, and it is not uncommon to see neutropenia in patients with X-linked agammaglobulinemia. And this, of course, will put them at risk of other infections. This diagram, I like it a lot because it is showing how BTK is involved in different areas, in different parts of the immune system. So BTK is linked to toll-like receptors, and toll-like receptors are receptors that are present in different types of cells, including the B cells, and they are responsible to identify microbes, bacteria, viruses, and fungi, whether they are present on the surface or they are present intracellular for viruses. BTK is linked with another molecule, intracellular, which is called, STING molecule, if you see here. And this is actually activated response to viral infection, nucleic acid recognition, and produces interferon. So BTK is important for viral production, so the response of interferon for viral productions. So it's not just about B cell, but it's also about other cells and linking to the toll-like receptor, linking to the integrin for activation of NK cells and monocytes. And also BTK deficient B cell actually fail to be an effective antigen presenting cell to the T cell. So going from here, as Angela and my colleagues have said, the patients with X-linked agammaglobulinemia are susceptible to enteroviral infection, and they can develop meningococcal encephalitis. Enterovirus is also a fecal-oral infection. The virus itself replicates in the upper airway and the distal part of the bowel. And in patients with X-linked agammaglobulinemia, there can be viremia and tropism to the CNS, inducing meningoencephalitis, as we can see here with MRI showing hyperintense lesions in the bones and medulla oblongata in this patient. And the main problem here is impaired production of interferon type 1 and 3 in response to the enteroviral infection. So treatment, so far there is no antiviral. They can handle this virus. We usually treat patients giving high dose immunoglobulin IVIG, 1 gram per kg, and frequency is variable across centers. There is no consensus. Some centers have also given intrathecal immunoglobulin therapy. Others have used subcutaneous interferon alpha 3 times per week. And some have given off-license intrathecal interferon beta. And prophylaxis, as I said, there is no clear consensus about prophylaxis against enteroviral infection, but some centers like Great Ormond Street Center, when I was working there, we were putting all the patients, keeping IgG trough level up, 10 or above to prevent enteroviral infection. How much this will affect, I'm not sure, because we know that it's mainly an impaired production of interferon rather than lack of IgG. Another pathogen that is quite important is oral polio vaccine. And in Africa, oral polio vaccine is given to, is a type of the vaccine that we give to our patients instead of the killed vaccine that is given in the Western countries. And the problem in handling oral polio vaccine starts from the very beginning. So I will go step by step. So first of all, in patients with X-linked agammaglobulinemia, there is lack of IgA, and this will allow the poliovirus to adhere and penetrate through the mucosa. And then, as I highlighted, there is an impairment. Step 2, there is an impairment of type 1 and type 3 interferon. And this means that the virus can replicate, and there is nothing that will clear the virus. There is a defect also in NK cell chemotaxis and dendritic cell maturation, so the virus can escape and reach the bloodstream. And when he comes to the bloodstream, the virus doesn't see IgG to try to clear the virus. So there is failure of neutralizing the virus, the circulating virus. So this virus will start going to primary organs, extraneural organs, heart, liver, and muscle, just circulate there. And then as the virus is replicating and there is nothing that is stopping this virus can revert to a neurovirulent virus and then go to the CNS and induce polioencephalitis. And saying this, I have seen patients. So I had a patient with X-linked agammaglobulinemia in Egypt, and he had the oral polio vaccine 246. He was fine, no problems at all, until he started to develop infections and stuff like this. And he was diagnosed around the age of 1 year with X-linked agammaglobulinemia. And then at the age of 5 years, this boy developed poliomyelitis. He developed typical poliomyelitis with paralysis, unilateral weakness. And when we looked at his stool, we found polio in his stool. And with more genetic testing, we know that this is the strain of polio. This is the vaccine strain, and it developed a mutation. So it's not that the mutation will develop immediately. The mutation might develop in years. So the polio is circulating until they develop this problem. So it's quite important to avoid oral polio vaccine in this cohort of patients. Other problem with X-linked agammaglobulinemia is autoimmunity. So 12 to 16% of the patient can develop arthralgia or arthritis, and 7 to 9% develop chronic diarrhea, and 3.4% develop IBD. So you need to be aware of this. And there is an increased risk of cancer. 1.5 to 6% of the patients, they develop colorectal cancer, gastric cancer with H. pylori, or lymphoproliferative tumor. We are seeing more cancer, maybe because they have chronic infection or they are surviving longer. So we are detecting this now. And this is a very nice study by the Italian team. This is the long-term follow-up and the biggest long-term follow-up study in XLA. And it has 168 patients. What I just want to highlight here is this is, so in on the left side, this is the time at diagnosis. This is the percentage of patients, and these are the different clinical presentations. And this is during the follow-up period being on immunoglobulin replacement therapy. And if you see here, so like almost like 20% had chronic lung disease at diagnosis. And if you go during the follow-up, despite being on immunoglobulin replacement therapy, they have 60% have developed chronic lung disease. And in this study, they clearly demonstrated in the figure Z that almost 50% of the patients in their study cohort by the age of 50 developed chronic lung disease. So it means that it's not, so despite being on immunoglobulin replacement therapies, they were still developing chronic lung disease, saying there is a problem with the BTK is beyond, it's not just the B cell defect, there is an innate defect, there is the absence of immunoglobulin E, and the IgM might have been affecting the outcome of these patients. And they looked at the mortality So this is in the x-axis, this is the age in years, and on the y-axis, this is the percentage of survival. The red is the normal Italian population, normal Italian male. And they looked at the whole cohort and they found 92% survival in comparison to almost 99% in the normal Italian male by the age of 40. And they found in the patients who had chronic lung disease, positive chronic lung disease, they have less survival than patients who have no chronic lung disease by the age of 40. So you see the survival drops from 98% to almost 90%. So I just want to highlight one thing about this study, because the age of diagnosis was 66 months. And they developed the National Italian Registry after the year 2000, so they can see that the age of diagnosis after the year 2000 have moved to 23 months. So I'm not sure if the outcome will be different in the patients born after the year 2000. This was not highlighted clearly in the paper. So how do we manage a patient with XLA? Again, we follow up the patient every 4-6 months in clinic. We initiate immunoglobulin replacement therapy. Centers are running IgG at 10. Despite regular replacement therapy, I'm saying there are patients having 50% can develop chronic lung disease by the age of 50 years. So lung function tests every 6 to 12 months, CT scan every year or every 3 to 5 months based on what is really happening. Avoidance of oral polio vaccine is an important thing. And it's a big challenge, but at least 1/3 of the X-linked agammaglobulinemia patient, they have a family history of X-linked agammaglobulinemia. So at least in this cohort where we can identify the patient, we should not be offering oral polio vaccine. And management of autoimmunity as per protocol. And then because BTK is not just about B cells, people were thinking about transplant in patients with BTK. And so far there are 8 patients who were transplanted from the United States and Japan. And there was no clear benefit of transplanting these patients, apart from one patient that I think is who developed the X-linked agammaglobulinemia and then developed AML at the age of 15 or 13 or 15. And the reason for transplant was mainly to cure AML. There is a potential of gene therapy, especially with the use of safe lentiviral vector, but I think the future is for gene editing and removal of this bad gene and putting the correct gene. Screening is very important, family history and Curex for B-cell defect, as my colleague has pointed out, Shuayb. And then the final part is more into CVID. So CVID is, as Shuayb has pointed out, is a constellation of clinical presentation. So most of the cases are polygenic, but there are some few cases, like 5-10%, that there is a monogenic disease responsible for common variable immune deficiency. It's not just a B cell defect; it involves T regulatory cells. It can include a T cell defect, and there is an impairment of dendritic cells. And the clinical presentation is variable, having infections, autoimmunity, granulomatous disease, and cancer. And autoimmunity is around 40-50% of the patients, and they develop autoimmune hemolytic anemia, autoimmune arthropathy, GIT diseases, other autoimmune endocrine disease. And maybe autoimmunity is the first manifestation, not necessarily that they will develop infections and then autoimmunity. Autoimmunity might be the first presentation of the patient. So you need to think of checking immunoglobulins and looking if they have common variable immune deficiency. So if there is autoimmunity, you liaise with the hematologist, rheumatologist, gastroenterologist, according to the specialty. And steroids are the backbone for treatment, short course, bridging therapy, I would think. Rituximab for autoimmune cytopenia, as we use for other scenarios. But if you know the disease, if you know the gene, in case of monogenic CVID, your treatment might change. So moving to what is called precision medicine or targeted therapy. And I will give here just 2 examples of targeted therapy for CVID. So one example is PI3 kinase gain of function mutation. So if you look at the picture on the top, so PI3 kinase is formed of 2 subunits, B110 delta and B85, and it is present as a transcription factor intracellular below the receptors, and is present on T and B cells and other cells, and is responsible for T cell activation and B cell activation and maturation and survival. So in patients who have, so once this transcription factor is activated, the patients will start to have, if you look at the diagram, will have AKT as a factor being activated, phosphorylated. I'm not sure if you can see my arrow, just it's going on and off. So AKT will be phosphorylated, and down there is the mTOR pathway, N factor kappa beta pathway, GSK pathway. So in order to manage a patient who have BI3 kinase CD, which is the B110 delta gain of function mutation, or BI3 kinase receptor 1, which is a gain of function mutation affecting the B85 subunit. We were thinking about how to inhibit this pathway to avoid the autoimmunity associated with this type of immune deficiency. So the first thing was to use an mTOR blocker like sirolimus, or the other thing is to use a BI3 kinase inhibitor. So this is targeted therapy. So we have a patient who has CVID because of this type of immune deficiency, and we are treating autoimmunity with a targeted therapy. We know where is the disease, we know where is the gene, and we are treating what we know. Another example is the example at the bottom. And this is another type of common variable immune deficiency, whether biallelic LRPA deficiency or CTLA4 haploinsufficiency. So when the T cell encounter the antigen presenting cell in order to stop the activation of the immune system after the immune cell has handled the infection, in order to stop ongoing immune activation, CTLA4 will go on the surface of the cell wall and stop the connection between the antigen presenting cell and T cell, and switch off the immune system from ongoing inflammation. So if this switch off button is not there, there will be ongoing inflammation, there will be lymphoproliferation, there will be granuloma formation. So patients who have CTLA4 haploinsufficiency or problem with the CTLA4 will have ongoing inflammation and autoimmunity. Another checkpoint for CTLA4 is called LRPA. This is something that is present intracellular, and LRPA is the one that put the CTLA4 on the surface of the cell, cycle the CTLA4 to the surface of the cell to switch off the immune system after it handled the infection or whatever thing that it was acting against. So if we have a biallelic LRPA defect or CTLA4 haploinsufficiency, patients will develop hypogammaglobulinemia, and they will develop autoimmune manifestations and granuloma. And to handle granuloma and autoimmunity in these patients, we can do targeted therapy by supplying the CTLA4 through the use of a humanized antibody called abatacept. So this is how we can use targeted medicine or precision medicine in management of autoimmunity linked to PID. Another problem with CVID is granulomatous disease. So they have activated and aggregated macrophage. 8 to 22% will have a granulomatous disease, and Shuayb has clearly demonstrated one of his patients who had this nasal problem and granuloma affecting the heart palate. It can affect the liver, it can affect the lung, bone marrow, kidney, skin, eye, and GI, and it is usually associated with autoimmunity, and it carries poor prognosis. And one of the things that I just want to highlight is granulomas that can develop in the lung. If you see the pictures, you can see there are like ground glass opacifications in the lung of the patients. And this has been clearly defined, diagnosed. Management is still not very clear by the British Society of Lung Foundation in the UK. And GLILD is a distinct clinical or radiological, pathological interstitial lung disease that occurs in patients with common variable immune deficiency associated with the lymphocytic infiltration. So it's not just macrophage aggregation, there is also lymphocytic infiltration and granulomas. They can appear in the lung. The most important thing is when we see this type of scans, we need to exclude other problems and in particular infections. So 8 to 22% of the patients develop granulomatous lymphocytic interstitial lung disease. Some of them will remain asymptomatic. CT chest is important at the diagnosis of a patient. Lung function test with gas transfer. As I said, exclusion of infection is a priority, mainly mycobacteria, fungal infection, atypical mycobacteria, BCP, viral infection. And in few patients, we need to do a lung biopsy to confirm the diagnosis. And in the lung biopsy, we usually look for CD20, the B cell, CD4, CD8 staining, and check for polyclonality and exclude lymphoma, which is increase in patients with common variable immune deficiency. And in terms of treatment, the first line of treatment is prednisolone, 1 to 2 milligram per kg per day, can be used as monotherapy. Second line option, as I said, the lung pathology biopsy can show B cell infiltration, so you can consider rituximab. There might be also T cell infiltration, so azacitidine and MMF as a lympholytic agents can be also considered. And third line option is infliximab targeted therapy. And in some cases, some centers will go for transplant in patients with GLILD. However, when to stop or need for maintenance therapy, so far there is no consensus. We know that the treatment is individualized because patients on immune suppressive medications can develop infections, so you might need to stop immune suppressive medications. We look at the whole picture. If the patients have other comorbidities like has diabetes, hypertension, and then we cannot give steroids, and a steroid-sparing agents is needed. There is no consensus yet about when to stop or the need for maintenance. And then the other problem that is there in CVID patients is liver disease, and it can range from nodular regenerative hyperplasia of the liver with immune dysregulation, infections, and liver cancer. It's quite important that we monitor liver enzymes, in particular alkaline phosphatase and gamma GT. We do liver ultrasound and the new technique, which is FibroScan, to look for fibrosis. I just want to say that in adult cohort, because I worked in Royal Free as well, and I can see in adults, we are seeing more liver disease in patients with CVID than what we see in pediatrics. And we can see a hepatopulmonary syndrome and liver failure in this cohort. Malignancy, as my colleagues have pointed out, so we can have lymphoproliferative lymphoma, maltoma, and again, H. pylori is quite important to screen for because they are 10 to 47 higher risk for developing gastric carcinoma. And prophylaxis, regular screening, the main thing, regular surveillance, and the treatment with the specialist team. And this is just one of my patients when I was working in Royal Free. I just want to share it with you. So he's a 56 gentleman who presented with renal calculi to one of his local hospital, and they did a CT scan and they found mesenteric abnormality. They said there is an abnormal thing in the mesentery. So we said, okay, we are going to do a PET scan. He was completely fine. Apart from that, we know that he had GLILD, and as you can see, the PET scan was showing uptake in the lung. Interestingly, we found an uptake in the thigh. We found an uptake in the lateral wall of his chest wall. He did not have any swelling or anything. He went for a biopsy and it was confirmed B-cell lymphoma. So I'm just saying that this was an incidental finding in B-cell lymphoma, muscular B-cell lymphoma in a patient with CVID. The final part of my talk is the vaccination. So for all patients with B-cell defect, there is no harm in giving all killed vaccine as per national vaccination program. It's important to give the pneumococcal vaccine Prevnar or pneumococcal 23 and conjugated Hib vaccine, and of course give the COVID vaccine. There will be talks about COVID vaccination and COVID in PID, in particular B-cell defect, in the conference. So probably you need to attend these talks. In terms of the live attenuated vaccines, so for patients with the selective IgA or SPED, we can give these live attenuated vaccines, but for patients with XLA or more severe forms of CVID, we need to just use the killed vaccines. And BCG vaccination, the risk and benefit, I think the risk of getting disseminated BCG is more than the benefit of preventing disseminated BCG is more than the risk of having BCG proper in patients with CVID. So we'll go with the BCG vaccination. And for the household, we give all the vaccines, MMR and varicella zoster vaccine and BCG vaccine. There is still a debate about giving live attenuated influenza vaccine or the killed vaccine. And this is just some of the recommended stuff in the literature that I think you can have a look at, discussing more in depth some of the information that I tried in this 30 minutes to highlight for you about management of patients with B-cell defect. Thank you.
Good afternoon, everybody. It has been a wonderful day. I have learned a lot, and I really thank all the speakers for their wonderful talks and the collaboration and all the hard work that has gone into organizing this and bringing talks in such a harmonious and well developed way. So thank you very much to Mirjam, to Reem, to Shuayb, to Hiba, to Angela. Thank you all. This has been a phenomenal day. It's been really great. Before I dash into discussing the questions that we have received quite a few. I would like to remind everybody to make sure that they have registered themselves on the website so that they can get their certificates for the conference as a whole after the end of the conference. To do that, I mean, that is also linked to completing an evaluation form for the workshop and for the conference. So after you register, you will receive the evaluation form. You can answer that, and then you will receive your certificates. For the workshop itself, there is also a quiz that will go live in a couple of days. You will receive notifications that it is on the website. So please make sure that you answer the quiz. And it is based on the lectures that have been presented today. This will qualify you to getting a special certificate on this module of B-cell defects. And we are planning 3 or 4 more modules in the coming 8 to 12 months to finish off a PID certificate for of the African Society for Immunodeficiency. So please make sure that you collect these certificates, and together they will make up your qualification. So this is a little bonus for everybody. So these are the key messages that I wanted to transmit, and now I ask you, Shuayb, to introduce the questions and let us know whom you want to respond to each one of them, please.
Thank you ever so much. It's been really an awesome day today. Thank you ever so much, Nahla, and all the speakers and all the attendees who is still with us today and posted their questions. We received so many questions. I tried to select the one will be more beneficial to everyone. And I would like to start with the first question to Mirjam, if you don't mind to answer this question. So the question was about the appropriate investigation in patients with immune deficiency. So the question about an adult, I thought from pediatric as well perspective, I'll start with that first, and then we can go to the adult perspective.
Yeah, that was then the investigation. Was it more linked to the CVID or more to investigations of antibody deficiency in general?
So the question was mainly about CVID.
Yeah,
if you want to expand more, it's up to you, Mirjam.
Yeah, okay. I think it's good to focus on CVID. I think for children, I think it's good that to realize that the CVID diagnosis could officially only be made after the child has been 4 years of age. I think that's important to as a starter. And then what diagnostics I would recommend now, first of all, the immunoglobulins and also the vaccination response, because that is also part of the classification and definitions. That's really crucial. And then you can perform a flow cytometry for further investigation of the B-cell compartment. However, it's important to realize it's also for children that's not part of the diagnosis, because of the variability of CVID, of the CVID spectrum, I would say, you'll have children with a seemingly normal peripheral B-cell compartment. However, the plasma cells in bone marrow don't produce antibodies. And it could also be that you have only really an earlier block, and then you have to see that you only see the earliest B cells. So that's the you can use it, but more at the most important steps I would recommend is the immunoglobulin and the vaccination response. And then the genetics, yeah, that's always a tricky point for that was also in your talk. So that's I think family history of where there are other complications, the chance of finding a genetic defect is higher. But I think it's really an important decision when and how to use it. Also taking into account you can face many other sequence variants and how you're going to deal with it. So especially for CVD, I think it's important, especially when you have more other non-infectious complications and a positive family history. I think then it's good to consider it. And otherwise, for these young children, I would be a bit hesitant or think before you order. You go to. I don't know how you think about it, so Shuayb.
Thank you ever so much, ma'am. This is very extensive, and I think it's quite challenging in the pediatric population. I think Hiba, I want to add as well something from her experience in UAE.
Yes, I think in UAE we get a lot of autosomal recessive conditions, and we see a lot of less profound combined immunodeficiencies, atypical kind of SCID. And I think it's really, really important. They can present with pure B-cell deficiency, bronchiectasis, recurrent sign of pulmonary infections. And then when you find that they have low CD4, so lymphocyte subset is something very important to look. Adding to Mirjam's is to look at your T-cell compartment, look at your B-cell numbers. If they're both low, you have low CD4s, you have, persistent lymphopenia, that should raise alarm bells that you could have a combined immunodeficiency.
Yeah.
Or if you have, other atypical infections, think about.
Yeah, I think it's a really good addition, because especially the T cells would be even more relevant to see in these patients than the B cell typing themselves, because if you have more indications for combined immunodeficiency, that would really also influence your management, I can imagine.
Absolutely. Thank you ever so much. I think this is really kind of covering the overview of how we investigate these patients' C-value. Yeah. Now, from adult perspective, Nahla, do you want to add something from that perspective?
Actually, I want to add something from the African perspective.
Go on.
I mean, basically, what's available to most of us is basic lymphocyte subsets. So what Hiba and Mirjam have just mentioned is quite relevant to think about whenever you encounter a patient with an antibody defect in whom you haven't demonstrated a secondary cause. So it's very important to look at your B cell numbers and your T cell numbers, and scrutinize your T cell numbers. You get the CD4 count, and we do see a lot of those CD4 lows amongst patients that we see. So, and most of them give would have like specific antibody defects in the ones in whom we succeed to do proper vaccination tests and that. So, so that's really good. That's really important. From the point of view of adults in general, again, and from an African perspective, we don't have readily available B-cell subsets. So the classification into naive, memory, and class switched is going to be quite tricky for African doctors at this stage in time. But and some centers are going more for genetic testing on panels at this stage in time for research purposes mainly. So it is a bit early for Africa, and we have to consider the limitations of what we have, but we have to think of moving forward as well.
Brilliant. Thank you ever so much, Nahla. And I think that's answered already another question about which flow cytometry panel we need to use for these patients' C-value. And you've already answered that. I think to start with, you're going to have the lymphocyte subsets, but then wherever we can, if we can get some sort of other phenotypes, that would be very, very important. Thank you ever so much. So there's a question here, is quite specific to Africa. So the African expert should answer that. Do we have any known centers of excellence in Africa that we can reach out to order or confirm genetic testing in patients with primary immunodeficiency?
Is that African expert me then? Okay.
Well, you, Angela, African expert.
Yeah, I think Angela was asking that question to flee away from the answer.
Yeah.
To escape answering it. So basically, I looked at that question and I sent a message to Monica Esser in South Africa. Well, I know the status in at least 3 countries of Africa. In Egypt and in Algeria, they are starting NGS panels. And they are doing them mostly for research. I'm not sure that whether they will be able to receive samples from abroad, but I am sure that this can happen. And I think this can actually be of relevance if we start talking about training, within Africa. I think that's going to be quite relevant. And now I'm going to read out what Monica has just sent me. So she said they have access to the Jeffrey Model sponsored InViti panels. So Jeffrey Model have sponsored panels for InViti, and they do limited kind of free tests for patients. So they can do that for diagnosis purposes, and they're also running research while West and whole exome and whole genome sequencing, basically. She said that they may be in the position to offer whole genome and whole exome sequencing to other countries in the future. But then this depends on the collaborations, on the established collaborations. At this moment in time, what really happens in Africa for countries who wish to do genetic testing is to send away abroad. North Africa has been quite successful in collaboration with European countries, especially France. And there's also a big consortium that Raif Jiha is having, and NIH is helping quite a lot. So collaboration is a key here, and there are some small buds coming up, sprouting in Africa. So we hope that things will move forward in other African countries. One last thing that I would like to add here, because it's something that really, I think it's important to highlight. That's the fact that most of the activity, the progress, and the movement forward with PID diagnosis and care has started in the countries which I have just mentioned, having started some genetic diagnosis, so North Africa and South Africa. So it's only natural that they are catching up with genetic diagnosis quicker than other countries. But more countries are now picking up more patients. So I am guessing that the drive and the case will be clearer to the governments and to their health authorities to help with introducing the necessary panels, at least, if they are needed. The good thing is that most of the time you don't really need immediate diagnosis unless you're completely stuck, or you really need clearer advice to consider for counseling and that. I hope I covered all aspects.
Thank you so much, Nahla. That's really a detailed answer. Thank you so much. Up-to-date answer. Now, we're going to go from practical perspective, and the question will be both for pediatric and adult. So for pediatric, I'm going to ask Mirjam and Hiba to talk about, and from adult, I'm going to ask Reem to answer this question. So this question is about if we suspect PID, especially CVID, sorry, we're talking about the vaccine response here. So how long after vaccination can we check for vaccine response for all the patients? So maybe from the pediatric perspective, do you use vaccine response as part of your diagnosis? And how long after you give a vaccine would you check the response for the vaccine in the diagnosis of antibody or B-cell defect? So Mirjam, if you want to kind of comment for that, followed by Hiba.
What I can comment is what in the Netherlands is often used is also pneumococcal vaccination, and also for diagnostic purpose. Mm-hmm. And then I think they measure at 28 days after vaccination, and they look at the increase of the certain subtypes. And I think the vaccination response is also always a bit difficult to compare between countries, because everyone uses own measures of positive, of good or poor response. And there are also publications about this dilemma, how to use it. but that's how we do it. And I think maybe, Hiba, you have other experiences. You use other vaccination as well to...
Yeah, I completely agree, Mirjam. We use pneumococcal for polysaccharide responses to look for T-cell independent responses. But this is proving a problem now with patients who've had Prevnar.
Yeah.
Because they've already had a conjugate vaccine.
Yeah.
So we haven't made any changes, but Shuayb said they're now using salmonella, which isn't part of the vaccination for polysaccharide.
Yeah.
And then we always compare with a conjugate vaccine, like, you know, tetanus or something.
Yeah.
Or Haemophilus, or tetanus is the most common one. And we would check 4 weeks later. And we tend to do it in patients above 2 years of age because, you know, the responses tend to be, they're not very reliable in the polysaccharide responses. Are naturally not excellent if you're less than 2.
Yeah.
I don't know if Reem had something to add as well.
Yeah, no, it's more or less the same. So in GOSH, we were doing like tetanus and response to Prevnar. And as you said, above the age of 2, we test pneumococcal. In Royal Free, they were doing in adult setting, they were doing total pneumococcal vaccine response, and they were testing the response to the serotypes of the Prevnar. So 7 out of 13 was considered the cutoff to say he has good response to this vaccine. But this is the Prevnar is just looking at the protein conjugate. So it's like tetanus. I didn't feel it's like we are measuring pneumococcal vaccine response properly as we should be doing. But we don't have, and Haemophilus influenza, some centers also check Haemophilus influenza, and in some setting they also check meningococcal vaccine response. So usually meningococcal vaccine response is checked in patients who have complement deficiency, just to see if they have antibodies against the meningococci or not, if they have some protection or not. But there is no difference. And we do, for example, for tetanus, we check the antibodies, and then like baseline antibodies, we give the tetanus vaccine and we check the antibody response in 4 weeks' time and see if there is response or not.
Thank you, Reem. That's brilliant. And obviously the salmonella, when I mentioned Hiba, is purely a research at the moment. It's not really practical yet, but we're trying to, especially we had a massive shortage of immunoglobulin. I think that's kind of worldwide problem. And we try to figure out which patient we may allow to stop them, especially a patient with specific antibody deficiency. So sometimes the only way you can try is to try a different vaccine to see if they responded to that or not, because pneumococcal and all that blava will be present in the immunoglobulin. So salmonella unlikely to be present in immunoglobulin products so far, but who knows? Do you want to add anything, Nahla or Angela, from African perspective in Sudan or Kenya? Do we do any different vaccine, or we do the same in Sudan and Kenya?
I'm actually who posed the question.
Oh, brilliant.
Because it's one of the challenges I face. And I know in my country, we still don't have the capacity to check for vaccine response. But hopefully with time and with the increase in awareness that we will be able to do this. Thank you.
Perfect. Nahla, do you want to add anything here?
Again, it's a difficulty with the measurement and sometimes with actually convincing the vaccination people to give these patients the vaccine and then checking the levels. I mean, the last patient I managed to get specific antibody responses on was like 2 or 3 years ago, well, 2 years ago. And we gave him the pneumococcal and the tetanus, I think. Yeah. And we couldn't check the levels at home. So they flew to Egypt over a weekend to get the tests done. So that's how cumbersome things can get, unfortunately. There are some people who would do them for research. It's not difficult, especially with the protein antigens. So yeah. So there are logistic difficulties more than anything.
Absolutely, absolutely.
But the principle is the same.
Brilliant. Thank you ever so much, Nahla, for that. So there's some practical question here for the pediatrician. So mainly will be Mirjam and Hiba. And that's with regards to the oral polio virus or polio vaccine. So we know from the presentations now, it could potentially lead to a life-threatening complication in children. So I understand obviously there already been a few recommendations in the West, but if Mirjam can expand a bit of what recommendation with regards to oral polio vaccine for children in the West here, and then if Hiba can add what we do in UAE and Africa, if you have got any kind of information about this.
Okay. Sorry, I have no experience with this, so I cannot comment on this question. Sorry for that.
Yes. No. It's just absolutely fine. And I believe they already stopped using that, Hiba.
Well, I think in Europe and the US, I think they stopped it on it's IPV. Yes. Actually, in the UAE, they give both. They're moving. They're giving both. They were on oral, and then they introduced IPV, and now they're moving away from oral polio. I think very soon oral polio will not be there. But it's something that we have to mention. Have to say, don't give oral polio. Don't give it. It's still available, but it's going to disappear. I think very soon.
Yeah.
I think I can comment on this, Shuayb, from Egypt, okay? Because we were with the WHO working on studies looking at polio vaccination in patients with primary immune deficiency. So we found that patients so we looked at the stool for patients for severe combined immune deficiency So polio vaccine, I would like to tell you first what is happening with the polio vaccine. So in Egypt, they knock on the door and make sure that every mom, every kid has the polio vaccine. So it is not that you need to go there. You don't go anywhere. They knock on the door. They make sure that you have the polio vaccine. They put it in the mouth of the child once you open the door. So it's like, it's very difficult to say, oh, wait and delay it for patients with immune deficiency. We tried with the health ministry when I was in Egypt to delay it like 6 months so that we know what is the status of the child. At least we know if he has an immune deficiency, but it's not working like this because they feel, no, this is a priority. And anyway, immune deficiency is rare disease, so it's not a priority for them. So what we saw is that, so I had a child with severe combined immune deficiency 8 months post, so they get the polio vaccine 2, 3, 6, okay? So he was 8 months diagnosed with severe combined immune deficiency, and he passed away from what was initially thought about encephalitis, but not sure what is the cause of encephalitis. And subsequently, the CSF and the stool was positive for the vaccine, the polio vaccine strain. And it was showing some mutations. It did not achieve the virulent mutation, but it was showing mutations. So probably it was linked to why she died of encephalitis, because the other viruses were negative. And another patient, as I said, the XLA, who had the polio, and then at 5 years, he had the virulent strain excreted in his stool, and he had evidence of poliomyelitis, normal poliomyelitis, as any other kid. And there is also another child, 15 years old, CVID, who had the same pattern as the patient with the XLA. So it is a problem for this cohort of patients with this type of severe forms of immune deficiency. But as I said, the countries the authorities do not feel the indication of holding polio vaccination, because, or even considering giving the killed polio vaccine as well, because what they want is to allow from the oral polio vaccine the herd immunity, and herd immunity is at the top priority of them, rather than giving the killed vaccine and protecting a small cohort of primary immune deficiency patients. So it's a big problem, actually. And I think it is the same across African countries.
Brilliant. Thank you so much. That's a good answer. Go on, Nahla.
Here in Sudan, they have started giving first doses of the live polio. We keep on getting outbreaks, and that leads to a similar situation to what happens in Egypt, but in bouts. So they go about the houses to vaccinate babies or children under 5 whenever there is an outbreak. The second dose officially is a killed vaccine now. So the first dose, they give the live polio. The second dose is a killed vaccine. And then they booster with with killed vaccine. So that's the current protocol, but it changes when there is an outbreak.
Thank you ever so much. There is a good question here about the newborn screen, and I know Mirjam have published a lot on that. So I just want to comment from Mirjam with regards to the West at the moment, the state of the art, and then if any of the African experts want to kind of talk a little bit about what the state of the art in Africa at the moment. Off you go, Mirjam.
Yeah. What I can tell from the European or the I think the European, the Netherlands, the Dutch perspective is that we have been starting newborn screening for SCID since January 2021 onwards, and we also did some studies on several aspects of newborn screening. And that includes also the parental perspective, because you confront the people that already early with the diagnosis. That's good if there's really transplantation needed or treatment needed. But there are also some false negatives or children with low T cells that restore afterwards, but that you would like to prevent. So there's a lot of developments going on and discussions how to improve it, that you reduce the number of false negative. And what I can also tell is that we just published, it was yesterday online, also some recommendations for how to use the terms well in the field of newborn screening, because the terms positive and negative can be used in either way. And also the classification of SCID and leaky SCID, atypical SCID, it was really, you could really make beautiful word clouds of everything was used. So that's, I think it's really a joint effort, because especially to picking up these children so early, it's really good to prevent them for getting infections. And there's another point I would like to raise also here in the context of newborn screening for XLA or B cell deficiencies. It is possible because technically you can measure the tracks for the SCIDs and the cracks for the B cells. But I still, it's not really convincingly demonstrated that you have a really clear clinical benefit from the diagnosis at birth. And that's one of the, you have this Wilson and Younger criteria. When are you allowed to screen for newborns? And then it needs to be demonstrated that you have clear clinical benefits. That has not been done if you do not screening. So early diagnosis is always important, but does that justify a newborn screening program for XLA? Because it's also costly, and especially for in crack screening, you will pick up a lot of false positives. And that's also a problem. So I think for XLA, it's easy to say SCID screening and go directly to XLA screening, but I'm a bit hesitant for that. And I think what we are now also starting up is a large international study to really demonstrate the clinical benefit from diagnosis at birth, or is just clinical awareness more important in the early diagnosis, and is really a newborn screening required for XLA? And that's also difficult because if you talk about individual patients, and of course you say newborn screening is preferred, but screening is also like a population-based screening is something else. And I'm also from the diagnostic field, so then you try to find a diagnosis for a patient. But when you talk to screeners for healthcare programs, for them, the population is so-called the patient. And what's good for an individual patient does not mean that it's also good for the whole population. And that brings you really interesting and important discussions. And I think for XLA, it's really important not only to look at individual patients. Of course, it's better it's diagnosed as early as possible, but is it also of benefit for the whole population if you look at the false positives? So I think this field of newborn screening is really interesting and moving, and it's good to discuss it on a global level.
Thank you ever so much, Mirjam. That's really a very detailed and concise and precise description about the SCID screening. I'm sure you've got, people know about your papers. You've been publishing a lot about this in the field of SCID screening. Nahla or Angela want to comment on the African state of art from the newborn screening perspective?
Well, I can say that at least in my country, we don't have a screening program in place. And I think a lot of it is pegged on what Mirjam was saying, is that the policymakers are not convinced of the burden of the problem yet. And even before we get to the policymakers, the healthcare workers on the ground are yet to be convinced of the burden of the problem. There's more awareness. We still have a lot of work to do. But as Mirjam said, we will screen and we find, and then what next?
Yes.
Because that's always the challenge. If I find the problem, what is the solution that I offer? I don't think I have an answer, but I'm still of the old school opinion. With a good history and physical examination, that should be the first screening point. It doesn't cost anything. We empower you with skills and we pick it up from there.
Thank you.
I think the schools you're organized are so extremely crucial that you create good awareness and good clinical doctors who think about these diseases. I think that's really important.
Absolutely. If you're not thinking about it, you're not going to test it. That's the main question. And I think, even in the UK here, we struggled with this for many years. more than 10 years, people asking about this newborn screen to be included. And until this month, we just started the trial, not even part of the national way. But it's how to convince the policymakers. The policymaker stakeholders need to come on board for this, and it's quite a political and diplomatic things, I'm sure. So moving on to other questions on complication now and how we treat this. So there's a question for Reem. Do you have any experience with trial of antiviral medication for chronic enteroviral encephalitis or enteroviral meningitis that's not responding to IVIG, or high density
immunoglobulins.
So what's your take on that?
So bucapavir was used as a capsid inhibitor. So it's one of the antiviral that was used for enteroviral sepsis in neonates. And it has been tried in adults in other enteroviral infection. What was seen in some of the trials is emergence of resistant strains. I haven't used it in any of the XLA patients, but I'm not sure if someone else have used it. Maybe I have missed to read the publications about it. But if there is an antiviral that can show efficacy in management, so I know that it is used for other enterovirus in neonates. I know that they have used it in polio as well in some patients. So it might be of benefit in XLA patients, but I haven't used it. I don't have personal experience of using it. I'm not sure if Mirjam has used it in any of her patients or Shuayb.
My experience, not with particular enteroviruses, but with norovirus and all these naughty viruses in CVID and XLA population. I think the main challenge I found with these patients is they cannot get rid of the virus. They just get hold of the virus and stay in the body chronically forever. Even with the COVID-19 virus, SARS-CoV-2, we have got a lot of chronic carrier here in Manchester, and we try to give them remdesivir and all the available monoclonals and all that, and still they hold to the virus. They just keep it in. They don't want to get rid of it. So I think it's quite variable between patient with CVID or XLA or other antibody deficiency, and I think it's quite a challenge. And until today, we do not have any consensus here in the UK, but perhaps other people have got their own experience and what they do.
I use antiviral off license. So I have used favipiravir nitazoxanide off license combination for norovirus. Some patients it works, some patients it doesn't work. I don't have clearly which patient is going to work on. I'm just trying it and seeing what is the outcome. But
when you come to the off license, if you use the ribavirin,
it's off license.
All
of them are off license.
Exactly. You used ribavirin for chronic norovirus, and if you used oral immunoglobulin for chronic norovirus, so the same immunoglobulin for orally, and it did work in some patients. So I think it's just kind of ad hoc experience. Anybody want to add anything here, or shall we go to the next question? So there's a question from Hiba about.
I think you covered it.
Yeah. Hiba, you've got a question about the conjunctivitis and XLA, is that right?
Yeah, I've seen it a few times, and again, one of the things that's very hard to treat. So I wonder if Reem has any tips for us.
No, I mean, we usually.
Do you know the pathogen Hiba that's caused the conjunctivitis and XLA in your patient?
It's most commonly encapsulated, most commonly something like Haemophilus or, encapsulated bacteria. But again, they're on antibiotics, it clears, it's off the antibiotics, it's back again.
I have got one.
So I've seen three patients. I've seen three patients, two brothers with XLA, and one was back in the UK.
I don't know. I mean, my experience is one patient had exactly similar picture, a lot of skin and conjunctivitis, and we found he had Campylobacter, and we had to admit them for meropenem for a couple of weeks until we cleared it, and he actually managed to stay a couple of months without it. But obviously, I think sometimes it's quite challenging. I don't know what Reem experienced with that.
Yeah, so I have seen it not in pediatrics, but in adults. So usually they have this recurrent Haemophilus influenza. I think it's mainly the pathogen is Haemophilus influenza conjunctivitis. And usually they have it not just here, they have it also in the lung. And with infectious disease, they were trying to use 3 months, changing antibiotics every 3 months as prophylaxis and using quinolones as well to try to clear it. But this is in the adult cohort. I haven't seen it as a problem in pediatrics, so I'm not sure if there is a difference in how they present, but I haven't encountered pediatrics having problem with conjunctivitis. It's mainly in other population that I see the problem with H influenza.
Yeah,
in particular.
I've seen something similar, but it wasn't with XLA. It was a CVID lady, and it was interesting that she ended up with granulomatous uveitis as well. And she was diagnosed to have a vitamin D deficiency. I mean, I'm just mentioning this story because that was a very vivid discussion that we had when I was in Newcastle many years ago. So basically, that lady had the granulomatous conjunctivitis, uveitis after, and on top of, her conjunctivitis. And then she also needed some vitamin D. And I think she ended up with a bit of intoxication at that stage. So it was a very unfortunate kind of situation. I think infection in the eye of these patients, the approach has to include the systemic treatment and reculturing every now and again. I think that would be one of the things. The other difficulty that we see with these patients also is the complication with blepharitis, which makes the anterior cornea even more scratched and more susceptible to more infection. I think it needs also a bit of collaboration with the eye doctors to see what would be the best, maybe, something to do with smoothing the cornea or something like that. I'm not sure. Perfect. But it is a very difficult one. I have to admit. Yeah.
Thank you. I'll just finish with the last question because I think we're very well over time. It's just a practical question. Yes. It's a practical question for our African expert: how accessible subcutaneous immunoglobulin across Africa? So if Nahla want to kind of give some comment on that, and then if you kind of conclude the workshop after that. Thank you.
Yeah, very limited, I would say. We are struggling with provision of continuous supplies of IVIG as it is, and subcutaneous is very limited in Africa. Again, in some countries of the north and in South Africa, but that's very, very limited. I think in Egypt they might have like 1 or 2 patients only on subcut at the moment.
True.
I have tried to convince one of the providers to provide one of my adult patients with subcut during the COVID pandemic because of, its ease for home therapy. We ended up giving him IVIG at home anyway, while it was available. And now we don't have IVIG. Yeah, so that's how things work.
Thank you so much.
It's very sad, I have to say. I mean, I'm saying this and I've got the, yeah, I've got a closing throat at the moment.
Oh, bless you.
Honestly, so many patients have died, and a couple have had progressive bronchiectasis because they don't have the IVIG. It's very sad, very sad. Anyway,
I think that's why we are doing what we are doing, Nahla.
Yeah,
building awareness, and Rome wasn't built in a day. This is a marathon race, and we have started, and we will soldier on. Yeah.
Absolutely,
but it's painful. Yeah. It's really painful.
To carry on. Absolutely,
yeah. Very painful.
I'm sure we'll get there. We'll get there at some point. It's this collaborative work, this global approach. I'm sure we will get at some point, and we'll get these patients with immune deficiency at a better position for sure.
Yeah.
Yeah.
Education is always a start. The more people are informed, the more they know, the louder the voice.
Absolutely.
The bigger the case. Absolutely. Yeah. The impact. Well, thank you.
Thank you, Nahla, for you to close the workshop now.
Thank you, everybody.
Thank you, everybody. This has been really great and impressive. I'm really happy that we've done it, and you made a great teamwork. Thank you. And thank you to all those who persevered till the last minute attending this workshop. And thank you to the patients and everybody who has added something to this. Thank you so much. Thank you. Okay.
Thank you. Bye for now. Bye.
Thank you. Bye, everyone.
Thank you.
Bye. Keep safe. Bye-bye.
Thank you. Thank you.